first sparks on quiet streets, two hearts finding where to meet. But life pulled us apart, left me talking to the dark. If I called you tonight, would it feel the same? Or have we both moved on to different names? Take me back to the start, before the quiet and the scars, when the world felt small and your hands felt warm. Take me back to the start, if only for a moment. I've been up since 3, scrolling back where we used to be. Half truths in this late night chat, both of us know.
Hello, hello, hello. Welcome to Let's Get to the Marks. I'm finally there. I know what I'm doing. I think thereabouts. On a bit early. I know, earlier than I said I would be, but there we go. Can't be helped. Gonna start with some warm-up stuff. So just some multiple choice to warm up with, just to get us warmed up, and then we'll get into the proper stuff. So yeah. I'll be going through biological molecules, all the usual stuff that's coming up. So yeah, exchange and transport and all of that.
Here we go. What?
Okay, cool. So, yeah, we've got some multiple choice questions. Let me talk you through these ones to start with. Gonna do some biological molecules through multiple choice a little bit here, and then, yeah, what can I say? I'll be going through everything, so don't worry. He says, maybe, potentially. Cellulose microfibrils are formed by hydrogen bonding between adjacent chains of alpha glucose molecules, bonded with 1 to 4 glycosidic bonds. Oh. This is about the correct statement about the polysaccharides of glucose. Amylose is a straight chain of alpha glucose monomers bound by 1 to 6 glycosidic bonds to allow for dense packing. Glycogen has a high proportion of 1 to 6 glycosidic bonds to produce a highly branched molecule for rapid release of alpha glucose. Amylopectin has a mixture of 1 to 4 glycosidic and 1 to 6 glycosidic bonds between beta glucose molecules for rapid release of energy. Well, that's pretty tough, isn't it? That's a difficult one. These ones I think are from the OCR syllabus just by their wording, but you know, not most of my stuff today is AQA, but you know, I do use, I'm not frightened to use questions from different specifications. So a group of students were given a 1% solution of an unknown digestive enzyme. They were also given 3 tubes containing an identical mixture of foods. The students carried out a different biochemical test on each tube before and after adding the unknown enzyme. The results are shown in the table. So this is before they added the enzyme. This one went purple. After they this one went blue black with iodine, and this one went brick red. So that's before. Then after they added the enzyme, this one is still purple. This one has now gone yellow orange, and this one is still brick red. Name the type of enzyme the students used. A, B, C or D. Is it protease, carbohydrates, lipase, cellulase? Now up here where my face is right in the middle of it, carbohydrates such as starch are made from monosaccharides joined together. Let me just label this so that you guys can, if you want to put your answers, if you want to be brave enough to whack them in the chat. Carbohydrates such as starch are made from monosaccharides joined together. Which of the bonds A to D joins monosaccharides together? So is it ester, glycosidic, peptide or phosphodiester? And then number 4, image shows an isomaltulose, a disaccharide formed from alpha glucose and fructose. So we've got alpha glucose, we've got fructose here with the kind of 5 points. Name the bond that holds the alpha glucose and the fructose together. Is it 1 to 6 glycosidic, phosphodiester, ester, or 1 to 4 glycosidic? Anyone got a clue? All right, hold on, peeps. And So, if you're looking at the first question, if we're looking at the first questione, do you know what I need to do? I need to go on to my, see if you can put them all up. So put your number with the question. I just need to make sure, actually, if I go on my thing here, I just need to make sure I've edited this so that you guys can actually make comments. Because I'm using an old stream thing. Ah, balls, I don't think I can edit it while we're in the stream. I don't know how often you lot can make comments. Sometimes on old streams I used to put like an 8-second delay in, which is not good. Let's hope I haven't got an 8-second delay. I can't edit the stream now while I'm on it. I can't edit it, I don't think. So that's a bummer. Never mind. Because I like a 1-second delay, but you know, I think I'm using an old GCSE stream where I used to have like an 8-second delay in case you got trolls and stuff on there. Anywho, let's think about these. So what we're going to go through, you're going to cancel out the ones. So a lot of this is going to be true and some of it's going to be wrong. So amylose is a straight chain. Yeah, that's true. Alpha glucose. Amylose is made of alpha glucose. But it should be 1 to 4, right? So that's wrong. Glycogen has a high proportion of 1 to 6 glycosidic bonds. That's true. Glycogen is highly branched, remember. Rapid hydrolysis to release energy. So rapid hydrolysis and for rapid release of alpha glucose. Yeah, I'm not sure about that. That sounds right. I suppose they want to rapidly release the alpha glucose, which can then be used for respiration. Yeah, I think C is probably the best answer. Amylopectin has a mixture of 1 to 4 glycosidic and 1 to 6. That's true. It is branched. Oh, yeah, beta glucose is wrong. Cellulose microfibrils are formed by hydrogen bonding between adjacent chains. Oh, it's beta glucose. So, yeah, it's definitely C, even by process of elimination. So, yeah, rapid hydrolysis, highly branched. So, well done if you got C. What about B? Who got B right? Who got number 2 right? We got somebody said C's 2, B's 3, B's 4, A, C, and somebody else has said D. So we've got a little bit of a disagreement on number 2, and then people are in agreement of 3 and 4. Let's check out number 2 because we got a disagreement on number 2, which is cool. That's what we want. So the trick here, right, is that look, we can see that. We can see for number 2 that it went blue-black before. It went purple before a positive protein test. Why would you get a positive protein test? Well, before you've added the enzyme, the unknown enzyme. Well, enzymes are made of protein, so we would expect a positive protein test before, and we would expect a positive protein test after because the enzyme remains unchanged. Oh, hold on a minute. Yeah, I suppose it might have had protein in there. Did they say they had the students carried out a chemical test on each tube before and after adding the unknown enzyme? Oh, sorry, the food must have contained protein as well if it went purple, but it didn't have. It had starch as well, so it had protein, starch, and it had sugar. Afterwards, we can see, look, what's happened. The starch has been broken down into most probably glucose, while we're still brick red there, or maltose, whatever. So yeah, it's going to be the unknown enzyme that they've added is going to be B. It's going to be a carbohydrate because the starch got broken down. So yeah, a little bit confusing with the whole purple business, but never mind. Carbohydrates such as starch are made from monosaccharides joined together. Which of the bonds A to D joins monosaccharides together? So hopefully you went B for 3. It's glycosidic bonds. And then over here, name the bond that holds the glucose and fructose together. Well, what did they call this? Alpha glucose. Remember, with alpha glucose, the hydrogen is above. So the hydrogen is above. So this is the alpha glucose. So that has to be bond 1. You've got bond 2, 3, 4. Normally you have 1 to 4 glycosidic link, and that is 5, and that is 6. So we know it's between 1, and then what is it on the old fructose? Well, I'm guessing that that is going to be 1 on fructose. Then there's 2, then there's 3, then there's 4, then there's 5, then there's 6. So it looks like a 1 to 6 glycosidic bond. I know, a little bit unusual, right? But I'm guessing we're going to go one, we've got two, three, four, five, six. So to the right, I think we number these sugars by numbering the carbon 1 as to the right of the oxygen atom. So I'm going to go with A, 4A. Everyone else go with 4A, or am I barking up the wrong tree? Obviously, it's not a phosphodiester or ester bond. It's a glycosidic bond. What do we go? Yeah, 4A. Nice. Nice. Oh my days. All right, let me get rid of the loser in the chat. Okay, we got a loser in the chat. Let me just get rid of this Risa Fleur. Yeah, you know, he's a bit of a bell piece. Yeah, what are we gonna do, eh? What are we gonna do?
What can I say next?
Yeah. We got a bit of a bell end in the chat, guys. Sorry, that stuff happens. So let me just get rid of this dude.
Get the
Beep out of here.
You have been destroyed.
Yep. And what we're going to do here, we're just going to give you a little time out, dude. And give me 1 sec. You know, if you're going to be a bell piece, you're going to get timed out. Right. Let's go to some more of these. Got a few more of these to get through. Sorry about that, folks. My daughter's got they're watching Super Mario something or other, and they had issues with the Super Mario. Right. Okay, what we doing? What we doing? Let's have a butchers. Let's have a little look. So where were we? Got lost there. As to where we were. We're doing some biological molecules to start with, and I'm just warming up with some multiple choice questions. The table below shows 4 biological molecules and their component elements. Which of the rows A to D correctly identifies the elements in each molecule? Now, this one's quite tricky. This is a tricky little number. Let's call this number 1. Let's call this bad boy down here 2. Let's call this 3 and 4. We're just going to do a few of these. Just to warm up, because I was going to start at 5:30, so I thought, well, let's just do a few multiple choice to warm up. So we've got 8 of these to get through. Okay, let's have a little look. Whoa, let's get the proper glasses on, eh? Um, I mean, the guy was right. I am a fatty, but still, you know, you can't have that dude showing up all the time. What we got going on here? Okay, cool. Don't know what that is. It's my WhatsApp pinging. Everything's pinging off. Right. Okay, so what we need to say is, what would the elements be in sucrose? Hopefully you guys know. I'm going to go through the answers now. You can put your answers up in the chat. I'll go through and then you can see how you're doing. Sucrose is my face. Oh, my face is bang over some of those. Let me just move the old boat race over here. 1 second, guys. I'm going down here while we... there we go. Okay, so you might not have been able to do that one. Number 3. So... get the pen over there. What we got? We got sucrose. What is sucrose going to contain? It's a carbohydrate. What do we know about carbohydrates? They only contain C, H, and O. So we're going to get rid of B right now because carbohydrates only contain C, H, and O. Hard bit's going to be, okay, cholesterol. Cholesterol is a lipid. What do we know about lipids? Exactly the same as carbohydrates, made of the same constituent elements. Let's get rid of A. So cholesterol is just a type of lipid, and lipids and carbohydrates all are made up of CH and O. So we know it's C or D. And then we come here, insulin. What is insulin? It's a hormone. It's a protein. Well, this is where it gets a little bit tricky, but yes, it doesn't, because we know we get disulfide bridges in proteins. So we're going to go with C, and you probably did. And then, yeah, you know, in ATPs, nucleotides, they've got a nitrogenous base and a phosphate group. So great. Let's zing D out of there. I mean, you do actually get some proteins with phosphorus in them, but not naturally occurring. But there are proteins that have phosphorus added onto them later in biological systems. So it's a little bit of a tricky one, but there we go. C for 1. Structure of this biological molecule, bish, bash, bosh. Which of the following options correctly describes the molecule? Is it a hexose monosaccharide glucose, hexose monosaccharide ribose, pentose monosaccharide glucose, pentose monosaccharide ribose? Oh, we need to count the carbons, don't we? So we got 1, we got 2, we got 3, we got 4, we got 5. So that is not 6 carbons, as long as my mind is not deceiving me and my eyesight. So we're going to get rid of A and B. And now, is this a pentose? Well, it's not going to be glucose, is it? It can't be glucose. Glucose has 6 carbons. So no, 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 no. It is a pentose. It's a ribose sugar, pentose. So hopefully you got the first 2 right there. C and, why did I put a tick? what an idiot. It's a D. And I'm going to get off pink in a sec, because writing in pink's a bit annoying. Which of the following molecules, A to D, would be a product of breaking a peptide bond during hydrolysis? So we're looking for the amino acid. Hopefully you know amino acids are held together by peptide bonds. So it can't be D. That's going to be a glycosidic bond. What is this? This is a carboxylic acid. So no, it's not C. There's a carboxylic acid group, and there's an amine group. What is this? Carboxylic acid, amine. Ooh, breaking of a dipeptide bond during a hydrolysis reaction. What's the difference between... Wee, sneaky little bummers. You've got to have good eyes for this. B is not a carboxylic, not an acid group. That is like, if you're a chemist, that's an aldehyde group. So yeah, it's A here for this one. I don't know why. Yeah, this is 4 down here. That was supposed to be 3, but yeah, whatever. The answer for that one's A. Sucrase is the enzyme that breaks down sucrose. Which of the bonds, A to D, is broken down by sucrase? Again, we've seen it all before, peeps. It's going to be glycosidic, so it has to be A or B. I actually can't remember. Is it beta or alpha? Sucrose contains alpha glucose, right? Yeah, unfortunately, this live shouldn't have ads, but when I set the live up, I use the GCSE one. Give me 1 sec. Let me see if I can change that. 1 sec. Listen, the advert money off of adverts, what, 2 pence? YouTube stick adverts on. Actually, you can actually withdraw everything on the live, but I can't get back into the setup now. Unfortunately, I can't get in. Otherwise, I'd also take off any chat delay as well. It should be low frequency, the ads. If there's any ads at all, it should be low frequency. But when I set it up, I had to choose a GCSE stream. Anyway, no need for me to explain all of that. Okay, what we doing? What we doing? What we doing? What are we doing? Let's move on, guys. So, do we want to do any more of these? Do you know what? I think I'm going to change tact quickly. I'm only warming up at the moment. We're just warming up. So see if you can remember all of the organelles. If you've got any questions about any of the organelles, please do put them there if you have any questions about any organelles. And then we're going to move on. So if you've got any questions about biological molecules or organelles, we're going then we'll move on. We still got to do water, nucleotides, loads of other things. So yeah, if you have any questions, let me know. I'm going to go through most of it today, hopefully. Hopefully we go through it all, but all right, all right. If you have any questions about the cell, let me know. Anyone got questions about the cell? I don't know, about like, what, 2 hours, 2 more hours? So I am going to go through exchange and transport. So the main things I'm covering are like biological molecules, cells, microscopes, exchange and transport, animals, plants, the immune system, biodiversity, basically everything that's coming up, right? Or maybe we look at some evaluation questions, some maths, or some 5 markers as well. I don't know if I'm going to go through in particular order, but I thought, well, let's have a little look. Does anyone have any questions about biological molecules or cells? So you've got here, I'll run through this if you don't know. Number 1, you've got the nuclear membrane. Then you've got the nucleolus, number 2. God knows what 3 is. I guess that's supposed to be a vesicle or a lysosome, but whatever. Yeah. The difference between a lysosome and a vesicle: lysosomes contain lysozymes, which are hydrolytic enzymes that break stuff down. 4 is obviously the phospholipid bilayer, your A-level. Try to use A-level terminology. 5 is the mitochondria, the site of aerobic respiration. You can see the cristae in there. 6, centrioles. They're not found in plant cells. They're found in animal cells, or in animal cells they make the spindle fibers anyway. And then which is used in mitosis, and they're also used, they help maneuver stuff around the cell with the cytoskeleton and that kind of stuff. And then 7 looks like it is, oh, they don't have all the organelles on here, so that's a bit crap, isn't it? 7 is supposed to be the Golgi apparatus, because of the way it's got like the C shape with the membrane bound sacs, and it has vesicles budding on and off. And 8 is supposed to be the rough endoplasmic reticulum. Does anyone have questions about any organelles? Otherwise I'll just move on. Yeah. I think the smooth endoplasmic reticulum, where is it? I don't actually know where the smooth is. Maybe it's around the back of the nucleus over there, I think, maybe, but maybe they didn't have it. They missed that one out. I think it's probably the Golgi 7, so yeah. No, logarithm. So in biology, we use logs when we're talking about the growth of like bacteria on the graphs where you have bacterial population, because when you start putting numbers down like 10 billion, that's really hard to plot on a graph. If you do it as the log, it's much easier to plot on a graph. So that's where we start using logs when we're doing massive population, and especially if it's an exponential increase, like they're multiplying every couple of minutes. Then how would you fit 10? How will you fit 1,000 bacteria at the start of your graph and 10 trillion on a graph? You can't go up in a unit scale from 10 to 10 trillion. You can't do it. So, but if you do the log, then it's much easier to do that, right? That's the basis, I think, really, of where logs come in biology. They don't use them a lot, but anyway, what I want to do is let's crack on and yeah, whatever. Let's go through some. Oh my gosh, let's do a little bit more. But is there any biological molecules or something? I don't really know. Let's have a look. Let's see what we can do here. Let's get rid of that. Oh, actually, what do you reckon? So, oh, jeez, Louises, man. Hopefully, you know you've got the electron microscope. You've got the transmission electron microscope for AQA. You've got the scanning electron microscope, the transmission electron microscope. Hopefully you can recognize that the black and white one here is from a transmission electron microscope. You get 2D black and white, and then scanning electron microscope, you're going to get like, it gives you the surface, blah, blah, blah. Make sure you know all of the resolution and magnification and what they mean. Like, you can define those. Just make sure you can define all of them. Hopefully you remember the stages of cell fractionation. Do you remember when you do cell fractionation, what kind of solution you need? Do you remember about how you prevent osmosis and parts of the cell exploding? So you need to know that. If you're on the AQA syllabus, you need to know this. If you're doing OCR biology, you don't need to know this, cell fractionation. So when we do cell fractionation and ultracentrifugation is when you spin it and the small bits come apart. Do you know which organelles come off first? Which organelles come off first? Which ones form a pellet? That kind of stuff. And do you know the conditions needed? And can you explain the conditions? So that's what you need to ask yourself. Yes, nice. You're right. The nucleus. So the first thing, we're going to spin it, and then as we spin it up to high speed, then as we slow it down, the heavier things come out. So we go up to a high speed, and if you think at a high speed, the light things like ribosomes are really light, so they're whizzing around fast. And as we slow it down, the heavy stuff comes off and forms a pellet. So you get a pellet like with your nucleus and all of that in. Now, why is it isotonic? Why do we use an isotonic solution? So when we split the cell up and we chop it up, homogenize it, why do we use an isotonic solution? Who knows? Yeah, I'll go for anti-parallel strands. Yes, very good, Daisy. No osmotic effect. We want no osmotic effect, which will cause damage to cell parts or make organelles swell up and explode. So we use an isotonic solution. Isotonic means the water potential inside the organelles and the cells is the same as the solution that we're doing the centrifuging it in. Why is it cold? Why do we use cold temperatures? Who knows why we use cold temperature? I'll go through anti-parallel strands. To prevent osmosis, but why the cold temperature? Yes, that prevents shrinking and bursting. Great, no osmotic effect. So that's all linked to isotonic, but why cold temperature? Yes, low enzyme activity. So we lower the enzyme activity. Excellente. There's one more thing as well, isn't it? We buffer, a buffered solution as well, so that it doesn't become acidic or whatever. We buffer it, don't we? Buffer, cold, isotonic. What was your question? Do you have to remember the specific speeds at which each organelle comes out? I bloody hope not. No, you don't have to remember the specific speeds. I will check for you. So you generally just go off in the heaviest like organelles coming off first and the lightest ones don't. What do you call the liquid that's left behind after you've got all the stuff in pellets? What do you call the liquid that's actually left behind? Do you remember what the liquid's called? I need my glasses on. Got to get the old the windows on. What you do have to know, actually, which I'll test you on in 2 secs. So the speeds. supernatant. Awesome. Boom. Nice. You got to know it's called the supernatant. Excellent. So the nucleus comes off, and then the rest is called the supernatant. And then the order that they come off is going to be nucleus first, then mitochondria, then the lysosomes, and then the ribosomes and vesicles. Ribosomes are the smallest thing, I think. So, yeah. And each time you spin, you get a pellet, but only one comes off at a time. So the first time you do the spin, the nucleus comes off. The second time you do the spin, the mitochondria form a pellet. The third time you do a spin, I don't know what's after the mitochondria. I guess it's going to be like lysosomes. It depends what is in there. You know, it's the next biggest one. I think you just have to know the rough thing. You don't actually have to know that. I'll tell you what you do have to know that I never remember because I'm not doing my A-levels tomorrow. But do you know the magnification of a light microscope? You have to know the magnification of the light microscope. Maximum magnification. Who can tell me the maximum mag of the light microscope? Nucleus is the heaviest. Use the ZU. What is the maximum magnification? Nucleus and then mitochondria. That's right. No, the max magnification is 1,500. So you have to know that. The magnification is 1,500. And do you know the absolute best resolution of a light microscope? You have to know the best resolution of a light microscope, limited by the wavelength of light, because a light microscope uses photons. The exact width of a photon, one light particle or one beam of light, ray of light, it's a wave after all, wave particle, whatever. Do you know the width of light? 200 nanometers. Thank you, Lily Newton. You can also call it 0.2 micrometers. Either answer is perfect. But the width of light is 200 nanometers or 0.2 micrometers. That is the point where you can distinguish two objects like two ribosomes as separate. It can't do better resolution than that. Electron is much thinner, so we can get a much better resolution. An electron can go to 2 to the minus 4 micrometers. So that is 1, 2, 3 more zeros, 1,000 times better resolution. So you need to know the electron microscope as an electron is 1,000 times smaller in width than a beam of light. You can get a much better resolution, about 1,000 times better. Now, the best electron microscopes with the biggest magnification, you need to know a transmission. They can go up to I don't know, 500,000 to like 1,000,000 times bigger magnification, but their resolution's about 1,000 times. Okay. The only issue with the transmission electron microscopes need to be cut thin because the electrons need to transmit, go through. Okay. Whereas the scanning electron microscope makes a false 3D image that's not a real image, but it's mapped by a computer. You know, the electrons bounce off the surface, and so they can be used on thick specimens, but there's slightly lower resolution and slightly less magnification than the transmission. Okay, what else? I am going to go through everything. Someone said something about DNA. Do you want me to go through the anti-parallel DNA strands now, or I can go through ELISA. Indirect and direct. Yeah, we can go through that. All right. Yeah. Let's have. Let me see what's on this. What's on this thing first. Oh gosh. Yeah. What else do I need to go about with cells? Let's have a little butchers, and we get on before I do. Before I get off of cells, we'll. Oh, I'm not doing this question. Sod that. Not doing that question. Oh, let's quickly take a look at a prokaryote. Just remind yourself about prokaryotes. What do you call the DNA? Obviously, you know they don't have a nucleus. What do you call the DNA? Chromosomal DNA, loop of DNA, free DNA. All of that. What's the difference between the DNA in a prokaryote and the DNA in a eukaryote? What's the difference? Who can tell me two differences? Obviously, I'll give you one of them. The prokaryote's DNA is not in a nucleus. What's the other difference? Oh, very nice. So check out that people not associated with histones. Histones are a protein. Imagine like a scaffold that holds the DNA together. And obviously later on when you do methylation and acetylation and methylation, that is like how tightly wound it is. What do we call DNA that is attached to proteins? Yeah. Well, chromosomes. Yeah. So, or chromatin. So, yeah, nice. This has got not bound to protein. It's free DNA. You got these pili around here. You got a flagella. Ribosomes. What's the difference between the ribosomes in a prokaryote and eukaryote? Who knows the difference between the ribosomes in prokaryote and eukaryote? Oh yeah, there's no introns. Nice. Well done. Yeah, 70S versus 80S. You guys are all over it. Nice. And then we got these tiny circles of DNA called plasmids. Tiny circles of DNA which are only a few genes long. And hopefully you remember the word by which bacteria reproduce. And oh yes, bacteria, prokaryotes, sorry, have a cell wall. What is the substance that's not cellulose or chitin or chitin? What is the substance? There's about 3 names for it that is in the bacteria cell wall. Murian, nice. Murian, yep, Murian. You can call it peptidoglycan if you like. Peptidoglycan. So it's like a peptide, meaning protein, glycogen. It's a glycoprotein. Murian or peptidoglycan, it's a glycoprotein. What else? What is the slime layer around the outside of the cell wall called? They've got like snot, mucus, slime all over them. Slippy. Next time you wash a chicken breast, if you're helping out in the kitchen and you're cooking, you'll feel the slime. It's called the capsule. Lovely jubbly. Don't get it mixed up with the capsid that's found in viruses. Awesome, 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 awesome. Well done, peeps. Well done. Right, this is just basic stuff, obviously. Let's get on with... Right, now... Oh, hello. What is going on here? Where have I flopped to? Let me see if I can give you some 5-mark questions on, well, at the moment I'm just covering biological molecules and cell structure. Okay, so the first bit of the stream is just biological molecules and cell structure, microscopes, the basics, the foundations. So let's see if you guys can handle a 5-marker on microscopes. Now this is a question that has come up on the AQA syllabus before. It's 5-marker. Remember, at the back of paper 1 there are 3, normally they're worth 15 marks. Sometimes it's a 6-marker, a 4-marker and a 5-marker. Sometimes it's 3 5-markers. So you've got 15 marks of just pure content. Now there are lots of YouTube biology people who recommend you doing that first, say it's a great confidence booster for you, and they say the worst thing, if you're someone who's crap at time management, get to the end of the paper, you're running out of time, you're, oh my god, and then you go, oh, I'll rush these 3 5-markers. Listen, if they're the ones you're going to bank 15 out of 15, or you're going to get 12 out of 15 on those 15 marks, start with them and guarantee yourself, you know, those 15 marks. Don't get to the end and have 7 minutes left to do your 3 5-markers and you only get half of the marks that you would have got all of them for. So contrast an optical microscope and a TEM. What the hell? Contrast how an optical microscope and a transmission electron microscope work. Contrast the limitations of their use when studying cells. So first of all, how do the two work? And what are the limitations of their use? This was worth 5 marks, and this came up in an AQA paper 1, a real one there. See if you can put it together. Give yourself 2 minutes. We're only going to do 2 minutes on these, but we're going to bang through a few of these on biological molecules and cells. 5 markers that could come up at the back of the paper for you. We'll have a look at some graph questions. Someone said anti-parallel DNA. I'll go. DNA is part of like biological molecules and that, right? So we'll go through a bit of DNA. Yeah, so biological molecules also includes enzyme action, proteins, lipids, which we've done a little bit of already. You've got DNA, RNA, DNA replication, water, ATP, inorganic ions, and then I'm kind of blending all of the first like four topics. I haven't started cell membranes yet, but don't worry, I'm going to get through all of it. Or I'm just going to lick the answers up for you guys if you're ready for the answers to this one. So both optical images but don't produce color images. TEM focused using a beam of electrons, but optical focus using glass. Least TEM uses electrons, but optical uses light. Optical microscope has lower resolution than TEM. Yes. What can't we use in a TEM? One of the things about the TEM is got to be a vacuum, right? Why has it got to be a vacuum? Electrons are so small, if they hit an oxygen molecule or a nitrogen molecule from the air, they're going to rebound. So a TEM must use a vacuum. This is a big problem if we want to look at something living like Daphnia. Now, Daphnia, you can do the heartbeat practical with a Daphnia, and you can look at it under a light microscope. You can't look at it under a TEM. Here's the answers from AQA. So let me move my fat head out the way. Let's move that up there. Bish, bash, bosh. TEM uses electrons, optical uses light. Boom! TEM allows greater resolution. Excellent. So you can see smaller organelles, and you could name one. Oh, you'll be able to see ribosomes. Remember, the light microscope, you can never see a ribosome with a light microscope. They're too small. You could see like a nucleus, chloroplast, mitochondria, maybe, cell wall, and that's about it. You can't see any of the smaller stuff like lysosomes or ribosomes. TEM is only, you can only see dead organisms with the TEM or dehydrated specimens. You can look at live ones with an optical microscope. TEM doesn't show color. Optical can show color apparently. TEM requires thinner specimens. TEM requires more complex, time-consuming. This seems to be all of the downsides of the TEM. But I would say the light microscope doesn't have as good resolution, which we kind of said in the opening one there, and it doesn't have as good magnification. All right, let's bus on to look at, I'm not going to do a plant one yet, or respiration one. Let me just look at okay, this is another 5 marker. I'm just doing biological molecules at the moment and cells, microscopes, the kind of foundation biology. That's what we're dealing with at the moment. We will go on to the other stuff. So describe how maltose is formed and where it could be attached to a protein to make a glycoprotein. This was a 5 marker. How easy can you get? I bet you guys will be singing if you get this. You'll be like whistling away in the exam. Well, yeah, OCR. Some of this is for OCR, but this will help you revise. It's just you won't get asked. You don't have a 15 marks of content at the end. The only thing I'm not covering, okay, so I'm not covering, if you're doing the OCR spec, you're basically doing biological molecules, cell structure. You don't have to do ultracentrifugation, so that bit you don't need to listen to. And I'm not going to be doing respiration and photosynthesis, which OCR people have tomorrow in paper 1. They have the liver, the kidney, photosynthesis, respiration, and the nervous system in their paper 1, which AQA don't have. So, whereas AQA, they do a little bit of biodiversity and that is all biodiversity and Simpson's index. So for OCR, the first part of this live is going to be useful to you because I'm doing biological molecules, cells, and exchange and transport in plants and animals in the first half. So that is going to be useful to you. Maltose is formed from 2 glucose molecules. Which type of glucose molecules? Good stuff though, Desi Pink. In a condensation reaction, which forms a glycosidic bond. Yeah, nice one. Good. Removing water molecules. Maltose is formed from 2 glucose molecules. The Golgi apparatus attaches. Oh, nice. So where could it be attached to the protein to make a glycoprotein? So what they're saying there is where, and someone in the chat cleverly put up the Golgi apparatus is where we modify protein. So that's where you're going to whack a sugar on to make a glycoprotein. Nice. Here is the mark scheme. Okay, it was only a 4 marker. My apologies, it was a 4 marker. It's made of alpha glucose. We get you a mark. Remember, it's got to be A level, so they want your A level knowledge. So don't say base sequence, say DNA base sequence. Don't say glucose when you can say alpha glucose if you have to. Joined by condensation reaction. Nice description of that, removal of water. Joined glycosidic bonds, which people mentioned. Hopefully you put 1 to 4, but does it matter? That doesn't seem to matter here. And it is made in, it's added to a polypeptide or a protein in the Golgi apparatus. You can call that the Golgi body or the Golgi complex. Right, let me just see. Got a couple more biological molecules for you. These are relevant for OCR and AQA. Explain how 5 different properties of water are important. This came up on an AQA biology exam, so boom. Right, yeah, I'm a bit hyper. I've just had coffee and I've got a chicken burger to eat here as well. So, excuse me, I might eat that at some point. These are the answers. Maybe I should have given you a little bit more time to do this one, but we want to get through all biological molecules and move on. So, a metabolite in condensation, hydrolysis, photosynthesis, respiration. So they're just saying it's a good solvent. What do they mean by metabolite? Well, think about it. H2O is needed for hydrolysis. We have to, if you don't have water, you can't hydrolyze a polypeptide into amino acids. You can't make starch that you've eaten soluble as glucose and move it around your blood. So we need water for its ability to hydrolyze big molecules. It's needed in the metabolism. It's a solvent because it is polar. It can dissolve amino acids and glucose, metabolite. So it's a great solvent because it can dissolve polar substances. All the useful stuff in your body, glucose for respiration, your transport substance has to be able to dissolve it and carry it. So it's polar. High specific heat capacity. People like to use the term it buffers changes in temperature. It means that if you've got a pond in your garden or you've got the sea, when the air temperature changes, the water doesn't change a lot. You'll know that if you step into the sea on a burning hot summer's day, the sea still feels freezing cold because the temperature will not change easily, which makes a stable environment. It's no surprise life started in the sea because we predict that because it has a high specific heat capacity, so it's a stable environment. A large latent heat of vaporization. This means when you sweat and when water evaporates from your body, it requires so much energy to evaporate water that it carries that energy away from your body and it cools you down. So the high latent heat of vaporization makes it a great cooling substance. Now, if we sweated alcohol, let's imagine we sweated ethanol. Well, ethanol boils at like 60 degrees or 70 degrees. You'd need to sweat twice as much volume of liquid, so we'd have to be a lot heavier as beings, which would be a lot of effort and energy wastage. So having water as a cooling substance, superb. Cohesion. So in order for water to travel up plants by mass flow, the fact that water molecules are polar means they can stick to each other. Cohesion. It also works in the blood and other things as well. So columns of water in plants, and you get surface tension, so little pond skaters and insects can walk across water. Okay, these are some of them. I guess we could talk about when it's frozen, it has a lower density in its solid state, which creates ice and insulates marine life. Okay, look, I know people are putting up in the chat things they want to go over, and I might be going a bit slow, so let me just zing through and just, is there any more biologicals? Nice easy one. Describe and give examples of chemical reactions involved in the formation of polymers from monomers. So that's really easy. Here are the answers for that if you want to get them down. So this is just a general one where you could talk about hundreds of different things. Talk about hydrolysis reactions, condensation reactions, give the correct names of bonds, the correct names of monomers and polymers, the right ones, right? So don't forget, lipids are not polymers. Okay, lipids are made of glycerol and 3 fatty acids. They're not polymers. They're not a big chain that goes on and on and on. They're just made of 4 molecules. Anything else? Anything else? Do people want to do the membrane? Is everyone cool with a membrane? Cell cycle graphs. Yeah. We're gonna. We will. Yeah, we'll go on to some of that. All right. Somebody said mass transport. I don't want to get too far ahead, but all right. Let me give you a quick 5 marker. Name and describe then, because somebody said membranes. Name and describe the different ways substances move across cell membranes. So I want you to come up with 4 different methods by which substances can move across cell membranes. Can you describe 4 different methods? Give me 1 minute, folks. I'll be back. All right, people. So what we got? I've got to make sure I don't touch this, and then nice. Here's the issue, right? The phospholipid bilayer will not allow polar molecules to cross it. So we know, first of all, we have simple, we don't need to explain here either. It says name and describe. So I would say simple diffusion of non-polar molecules, things like carbon dioxide, oxygen, kind of like they can just zing through, right? They can just straight through. So small non-polar molecules can go through by simple diffusion from a high concentration to a low concentration across the membrane. Give an example. Then you want to talk about maybe facilitated diffusion, which is for slightly bigger molecules or for polar molecules that have to go through a protein channel, a channel protein. And then you can talk about active transport using a carrier protein, and that uses ATP to ADP PI, but we don't have to over explain, but that's against the concentration gradient. And then of course somebody has mentioned co-transport like sodium and glucose. And then of course we have exocytosis, which is where a membrane-bound sac, a vesicle, because the vesicle is covered in, you see these phospholipids, the heads with the little tails, the vesicle is surrounded by those as well. So when the vesicle, its membrane meshes, it becomes one with the cell membrane. The vesicle disappears, it becomes part of the cell membrane, and it releases its contents into the outside. When you do endocytosis, when you bring it in, the vesicle that's coming in becomes one with the membrane. Its phospholipids just move out into the membrane. So they're slightly different. Anyway, here is the answers for 5 marks from AQA. Simple diffusion of small non-polar molecules down a concentration gradient. Facilitated diffusion down a concentration gradient via a protein channel or carrier. I mean, we normally say channels for facilitated diffusion. There is a situation where you can use carriers as well. I'd stick to channel. Osmosis down a water potential gradient. Someone mentioned that in the chat. Active transport against the gradient via a protein pump or protein carrier using ATP. And co-transport, you could name 2 substances, sodium and glucose, which is part of how glucose is absorbed in the small intestine. You have to do that when we do digestion and absorption. So we get on to that. Okay. So well done. If you got this now tomorrow, you're going to smash it. Okay. What else? Let me have a little look about what else. Give me one sec. That's more. So, okay. What else? Okay, I think I might move on to a bit of DNA and RNA and stuff. So we can move on to, or shall I, do people want to do more biological molecules, or do you want me to move on? Do you want to do like, are you okay with why amylose is a good storage? Why amylose is good for storage? Amylose is compact, only got 1 to 4 glycosidic links, straight chain, coiled, and it's insoluble. Remember, all of the storage molecules are insoluble, so they do not have an osmotic effect. So amylopectin is highly branched, so it can be hydrolyzed. You can still say compact, takes up less space, less volume, and yeah, is insoluble. Glycogen, even more highly branched because animals need to energy faster than plants. So glycogen, even more highly branched, 1 to 4, 1 to 6 glycosidic links. But you can still get away with saying glycogen is quite compact, so it takes up less space when you store it, even though it's not as compact as like amylose, a straight chain carbohydrate. But remember also, all of these are insoluble, so they don't have an osmotic effect. So if they're asked you why they're good storage, Yeah. Yeah, there's two different versions of starch. So, Gallophius, there's two different versions of starch because amylose is for more long-term storage. You can't break it down as quickly, but you can pack more into a small space. So it's all about balancing. I mean, if it was only amylopectin it had its hands on, it can break it down quickly for respiration into alpha glucose, but it would have to take up more space, which means the plant would have to grow bigger roots and be bigger against gravity than it needs to be, and that would have to balance that against how much ATP and molecules it's got to use to get bigger. So it's all a balancing act in nature. So it has some long-term storage amylose, which is slower to break down, but it can coil like a spring and compress and store in a smaller space. Yeah. What else? What else? What else? Those are the two types of starch. Yes. What else? What else? Yeah, I'll get onto some of those application questions as well. Um, all right, all right, all right. Hold on. Let's get onto some application. Well, no, I'm gonna still go through a bit of content. What are we doing here? We're over here, and if I take this down and I take that down, let me see what comes up. Give me two shakes of a cat's tail, people. I'm just messing about. All right, so it's gone black. That is not what we wanted. So how do I, let me zing over here and how does that work out? Oh, I've got a black screen. Lovely. Okay, so why can I not see that then? Why is that PowerPoint not showing? Oh, this thing can be so annoying. All right, what about this? Let me see this. Give me 2 secs because I have to see what is coming up. What the no. Okay, at last. At last. Don't know what's going on with my techno's ganging up on me. Someone early on in the chat asked about this. I will get into DNA. Someone asked early on about this. So I'm just having a look through various slides and PowerPoint. Someone asked about this earlier. So, um, what do we want to do? Maybe I should go a little bit back. How far are we going back? Way back. All right, let's get to this. We're going to go through DNA and nucleotides. So I'm going to go through DNA and nucleotides. I will get to the 3-5. DNA and RNA are polymers of which monomers? So nucleotides, deoxyribonucleic acid and ribonucleic acid. What is the base structure of a DNA monomer? What is the structure of an RNA monomer? So you may well be asked this. So I'm just going to go through nucleotides and DNA, and then we will move on. So you're going to have a phosphate, and this is the easy peasy drawing, right? So we're going to have a phosphate group. We're going to have deoxyribose, which is a pentose sugar. If they ask you a similarity, they're both pentose sugars. Obviously, this has less oxygen. That's the one difference. And then do not call this just a base. Try to call it a nitrogenous base. And then the structure of an RNA monomer, it looks very, very similar. So just be saying to yourself, do I know the similarities? They both got phosphate. This time it's just ribose, not deoxyribose, but it is, oh my days, it is a pentose. Don't check out my Sugar Ray writing. It is a pentose, and then of course it's got a nitrogenous base. So if we were looking at similarities, they both contain nitrogenous bases. They both contain pentose sugar, and they both contain a phosphate group. However, what's the difference in the bases, guys? What is the base? How low can you go? The difference, oh my days, what's happened there? All right, the difference is these can be A, T, G, or C, right? And these can't be A, T, G, or C. What are we missing? We got U, G, C, or A for these bad boys. There's no thymine in the there. So you should be able to smash out your differences. Another similarity, right, is they no, that's loud. Other similarity, yeah, they both go blah, 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 blah. Move on. How are nucleotides joined together? Same as every biological molecule. How are they joined together, guys? When we form the polymer RNA, how are they joined together? What holds the strands together? These are two different questions. What joins adjacent nucleotides together, and what holds the strands together? Well, maybe not. Anyway, let's not worry about that. But essentially, I think what we're trying to get at here is that we have phosphodiester bonds holding these bad boys together, right? So we have phosphodiester bonds. Now, phosphodiester bonds is this bond here. So make sure you know that phosphodiester bonds hold. That is called being adjacent. These two are adjacent to each other. They're next to each other, neighboring. But opposite, what holds opposite strands together? Obviously not in RNA, but actually, yes, in RNA, because when mRNA binds to the ribosome, or when tRNA binds to mRNA, what holds two opposite strands together? Well, that is going to be hydrogen bonds. So just make sure you know, read the question carefully, RTFQ, whether they're talking about what holds opposite nucleotides or adjacent nucleotides. So opposite in two strands is hydrogen bonds, and adjacent is phosphodiester bonds. Awesome. Oh my gosh. I will get on to doing questions, by the way. I'm just going through some of the content. I'm going to go through as much as I can. So, yeah. Okay. What type of bond is between the complementary base pairs and adjacent nucleotides? You notice that's just linked to what I've been going through. So what's the order? What are my two answers, peeps? And then there's some maths. I don't want to hammer on with the maths. You will get like 10 marks of maths, but you know, I will do a little section on maths. I'll do a section on maths at some point. I'll go through separately tonight. If I've got time, I'll do 30 minutes of maths questions. Good, great. It's going to be hydrogen bonds here. This is going to be H bonds. Don't write H bonds in your exam. Actually write hydrogen bonds. And this one is going to be phosphodiester between adjacent. So just RTFQ, make sure you read the question. Describe the structure of DNA. 5 marker. So if you had to describe it, I'm not going to go through that. Easy peasy. Just make sure, do it in your head now. Can you say 5 things? Great. Describe how a phosphodiester bond is formed within a DNA molecule between two nucleotides. So what's the name of the reaction and what gets removed? And which two parts? So what they're asking you is, in this question, think about this. I mean, again, this is just content, but they're basically asking you, how is that bond formed? Oh my God, what was that? That was supposed to be a base. Okay. Bit crusty. But they're asking you how that bond is formed. So what type of reaction? Like all biological molecules, it's going to be condensation with the removal of a water molecule. And it's a 2 marker. I want to say 3 things. So I'm going to say between the ribose, deoxyribose on one, is it DNA? It's DNA. So between deoxyribose on one molecule and the phosphate group on the adjacent molecule. So I'm even describing the location just in case. I want to give the examiner 3 things. Okay. And then this comes, we got the 3, 5, 5, 3. I am going to go over the 3, 5, 5, 3 in a minute. Okay. Oh my God. I have to stay awake. Drink Fanta. Draw a box around a single nucleotide. So that's dead easy peasy. You might be asked how many. I've seen little questions which say how many nucleotides are here. So you just add up all of these. This is a nucleotide. Notice these ones are upside down. It's because it's anti-parallel strand. We'll go over that in a sec. The table below shows the percentage of bases in each of the strands of a DNA molecule. Complete the table by adding the missing values. So if strand 1 has 16% A, what does A bond with? So if 16% is A on strand 1, what does A bond with? A is going to bond with T, adenine and thymine. So that means the second one has 16% thymine. See, a little bit tricky, right? Then if that's 21% cytosine, what is so this one, let's say C is 21%. That means that G on this one must be 21% because they pair up. So a little bit awkward, this one. And they've all got to add up to 100, I guess. And then that's 34% G. So that means that this will have 34% C because you've got to have the same amounts of G's and C's on the opposites. And then I guess we just got to do subtract from 100, right? So whatever that is, 50, 71. I can't add up. So I'm going to go, which isn't great, is it? What's that? 44, 50, 71. Does that leave 29, 71? Yeah, I think so. Everyone cool with that? That one was quite tricky, actually. Okay. Quick reminder, quick reminder. Don't get your semi-conservative replication mixed up with your protein synthesis, with your translation and transcription. So in semi-conservative replication of DNA, this happens during mitosis, before mitosis and before meiosis, in the interphase, in the S phase, the synthesis phase. This is happening in your S phase. This is the doubling of the DNA. And how's it done? Helicase, the enzyme DNA helicase, breaks the hydrogen bonds. So you got to know that. Then the bases match up by complementary base pairing. Now, DNA polymerase, don't say RNA, it's DNA polymerase in DNA replication. But when we're doing translation and transcription, it's RNA polymerase. So DNA polymerase joins the nucleotides together by making the phosphodiester bonds, by creating the sugar phosphate backbone. Okay. And then hydrogen bonds form between the strands. And so just make sure when you're revising tonight, you're like, right, if I get DNA replication, just make sure on your hand you can tick off 4 things and then 4 or 5 points. Then you know, boom. As long as you can, every single thing, you can tick off 4 or 5 content points, you're sorted. So why am I flicking about? So just make sure you know that. And then here's like, the enzymes DNA helicase and DNA polymerase are involved in DNA replication. Describe the function of each. So if you've ticked them off on your hand and you know this off by heart, you're going to be like, DNA helicase breaks. Obviously, don't do what I'm doing. Write the word hydrogen. Breaks hydrogen bonds between opposite strands, or between, I'm going to go extra. I want to show off to the examiner I want to show off that I can't spell nitrogenous bases on opposite strands. I'm just making sure it's really clear the examiner knows that I know what I'm on about. So DNA helicase breaks the bases, breaks the hydrogen bonds between the bases on the opposite strands. Okay, if we want, some people like unzips the strands, right? The DNA strands. So whether you want to put that in quotations or whatever they call it, I don't know, whatever that is. Right, DNA polymerase has a different job. So DNA polymerase forms phosphodiester bonds between not opposite but adjacent nucleotides. You might want to say, oh my god, that's disgusting, that E. Look, check out that E. You might want to say it forms the sugar phosphate backbone. You can say it moves down the DNA You could give the direction if you want and all of that business. It moves down along the template strand, helping with the attachment of complementary bases and then forming the phosphodiester bonds between the adjacent nucleotides to form the sugar phosphate backbone. Right, what else? DNA polymerase moves in opposite directions along anti-parallel strands. This bit confuses some people, so I'm going to go through it now. Some people were asking about this earlier. So DNA polymerase moves in the opposite direction along anti-parallel strands. Will this come up? Is this going to make or break your exam if you fluff it? No. But so don't kill yourself over trying to understand it, but I'm just going to go through. So DNA polymerase, the active site of DNA polymerase is only complementary to the 3' end. And here's the tricky bit. This is less likely to be asked. It moves in the 3-5 direction along the template strand, but it is a bit of a lie. It's not actually attached to the template strand. What I would say is better than what is typed there is DNA polymerase, and this is important. So this is, if I had to remember two things about the direction of DNA, this is what I remember. DNA polymerase reads template. That's the original strand. Oopsie. DNA polymerase reads template strand in 3 to 5 direction. But I would say more common to be tested is the second bullet point, and that is in which order are these being made. Now remember they're anti-parallel. So my purple one here, this purple bad boy here, let me just rub this out, right? Let me rub these out and rub that out. So, yes, it's reading in the 3 to 5, but the new strand is always built in the 5 to 3 because the new strand is going to be like this one over here. That's not the template. It's going to be in the 5 to 3. So the new strand is going to be added on in the 5 to 3 direction with each nucleotide being in the 5 to 3. So what we want to say is DNA polymerase constructs the new strand in the 5 to 3 direction. So it reads in the 3 to 5 and the new strand is built in the 5 to 3 direction. So DNA polymerase builds new strand in 5 to 3 direction. So each nucleotide is added on in the 5 to 3, 5 to 3, 5 to 3, heading in that direction, because when this new strand is eventually built and as it moves along, we're not going to talk about Kawasaki fragments and all of that because of the split forks and all of that. I don't think you've got to know all the split fork business, so don't worry about that. As it adds on, this is going to be 5 and this is going to be 3. Yeah, so it's making the new strand in the 5 to 3, but it's reading the template in the 3 to 5. Anyway, that's all you want to go. Don't overkill yourself on that one. Does the DNA polymerase do anything with the complementary base pairing? It's not going to harm you to say DNA polymerase allows, not really, because you don't, they're going to line up by random complementary base pairing anyway, because as these molecules are moving and colliding mathematically, they're going to bond in that way anyway, whether the DNA polymerase was there or not. But you can say, oh, DNA polymerase aids in it. So this is just showing you this strand is being read in the 3 to 5 and being attached there. Remember, there are 2 DNA polymerase. The other one is on the other template strand, reading it also in the 3 to 5, but attaching it in the opposite direction. Great. Now what I don't want to do, and then of course with the new strand, you see here this purple one is the new strand, 5 to 3. So it forms in the 5 to 3, the new strand, and then the hydrogen bonding appears. Now depending on your syllabus, you might need to know if there's 2 hydrogen bonds between A and T and 3 and G and C, whatever. Yeah, I don't know, like, is everyone cool with the heavy and light nitrogen practical? Is everyone okay with heavy and light nitrogen? So essentially we grow some bacteria for several generations in DNA that only has light nitrogen. This is the evidence. So I'm going to just give you the brief one. There's no point in getting deep on it. Just to give you the brief one, we grow for several generations, and we'll do that as red. We grow the bacteria in light DNA, light nitrogen. Now, light nitrogen is N14. Heavy nitrogen is going to be N15. Now, I'm simplifying it. I know nitrogen has 2 atoms in it and all of that business, but we don't want to overcomplicate. So we have light and we have heavy. What do we mean by growing it in nitrogen? We mean the amino acids and the proteins that the bacteria have access to incorporate in their body, and therefore the protein where they get their nitrogen from to make their DNA is only light. Now, you do that for several generations until eventually all of the bacteria only have light DNA. If we extract their DNA, doing the DNA extraction, and then we spin it in a centrifuge, we find that the light DNA is going to like appear there, right? Now, if it was not semi-conservative replication, if it was conservative replication, which it's not, what would happen is when we take the bacteria and then introduce them into heavy nitrogen, what would happen is we would get this. So this is the incorrect way. We would get heavy and we would have light, right? That would be after the first division. We'd then allow them to divide again, and we'd look at their, we'd then centrifuge out, and what would we expect to see? Because we're only growing them in heavy nitrogen, we'd expect this to get thicker, and this band would only stay the same because this generation, their DNA is staying the same. This would be conservative. This is not what happens. So with conservative, with each generation, this band will get thicker because conservative says that the original DNA stays together and that it's just copied and built. That's not what happens. So what actually happens is semi-conservative replication. And what we find is instead of getting that there, we find we get hybrid DNA that floats when we centrifuge it somewhere in between, like this, oopsie, and like that, right? And actually, there is none light left. That band disappears. The light band is gone forever. So the light band that was on the initial one is gone forever and doesn't come back. Why? Because it's all been incorporated with heavy in a hybrid. And then what we would expect to see if it is semi-conservative is these are going to split again, and we're going to get more hybrid because you're still going to have your original 2 reds, and those original 2 reds are going to join with 2 new strands that have been made. But the thing is, the black strands here are also going to be separated and red, and they can only make black now, these heavier ones. So we're now going to have double, and that's going to keep getting heavier and heavier. So on this generation, you get the heavy appear and the medium, and then from then on, it's just going to be the hybrid's going to stay the same because you've only got that initial light DNA, so that's going to stay in the middle, and the band at the bottom is going to get thicker and thicker and heavier, and it's just going to get not heavier, but that's going to get thicker and thicker as you keep getting more of that with each generation. Anyway, that was the evidence for the semi-conservative replication. So before we get off of DNA, oh my gosh. How is DNA stored? Yeah, plasmid, circular DNA, not attached to histones. We already went over that. How is DNA stored in eukaryotic cells? So in eukaryotic cells, it's linear, thread-like structures, chromosomes inside the nucleus, wound and bound to proteins called histones. A chromosome, if you're asked to describe a chromosome, it is DNA attached to a histone. So here's a 3 marker. Very quickly, what are the 3 differences between DNA in the nucleus of a plant cell and in the DNA of a prokaryotic cell? And so what is the difference? I guess we can't say in the nucleus, but we can say like bound to histones. No histones. And then DNA in animals and plants and eukaryotes, fungi and protists is linear or straight chained. I know it's in an alpha helix, but it's still considered straight. And this is known as circular. Now, I can't think of another difference. What's another difference in the DNA? What's another difference? DNA in prokaryotic. Someone come up with the third one, because I'm losing a mark from my A level here. Longer versus shorter, like it. No exons, thank you. Introns, yeah. That's a good point, actually, that you do sometimes get some circular DNA, and that might be a little trick question for you. So, but that would be in the chloroplast. So we could say linear oh, it says in the nucleus. There we go. You're right. You're right. Whoever said that in the plant section, that plants have some circular DNA floating in their chloroplast, but not in their nucleus. So they made that specific, actually, that it's in the nucleus, it's linear, whereas that one is circular. Cool. And then longer, shorter, and then some other people have put some others about exons and introns up there as well. Nice. Yeah, absolutely. They have circular DNA, mitochondria, and chloroplasts. So, DNA code is arranged in threes called triplets or codons. Remember, each triplet codes for an amino acid. So that's codon, that other name for a triplet of bases. DNA contains non-coding DNA. Prokaryotes have exons only. They have no introns. They do have some functional RNA, though, apparently. Eukaryotes have exons, so coding DNA, and they have introns. So define the term exon, a section of DNA that codes for what? Genes. And this is just going through, yeah, exon, intron, exon. Okay. What else? Scientists, let's see this two markers. Scientists investigated the genetic diversity between several species of sweet potato. They studied non-coding multiple repeats of base sequences to find non-coding base sequences and describe whether non-coding multiple repeats are positioned in a genome. Do you have to do that for paper 1, or is that more paper 2-ish? So we've got non-coding base sequences. What are they? They don't code. They're the introns. They don't code for what? Structural proteins or functional proteins. And where do the non-coding multiple repeats are positioned in the genome? Where do we get those like CAG things? Let's have a little butcher's hook. So, oh my gosh, I've got the answers here, but I need to cut and paste them. DNA that does not code for proteins, DNA that does not code for a sequence of amino acids. This is all for the first mark. DNA that does not code for tRNA or RNA. Ignore reference to introns. So even if you said they're called introns, they're saying ignore. They're not saying you're wrong. So when you see ignore on a mark scheme, that doesn't mean you were wrong for writing it. It just means that's not what they were awarding the mark for. 2, they're positioned in between genes, except at the end of chromosomes, or positioned in the telomeres, the end bits, the bits that get shorter as we age. Right. Alleles are genes which code for the same characteristic, although they have slight variations in the nucleotide sequence, Alleles are found at the same place on homologous pairs of chromosomes. What is a homologous pair of chromosomes? What is a homologous pair? Name the fixed position occupied by a gene on a DNA molecule. Do you know the fixed position occupied by a gene? Like these two lines are in the same place on that DNA. And what is a homologous pair of chromosomes? Do you know how to define those? Oh, got 100%. Let me put that there. Well, the position is the locus, but what is the homologous pair? 1 second, people. So what do we reckon? Where are we? Sorry about that, folks. Just a quick reply. Same genes with different alleles. So you have to be a bit more specific because it's the here we go, HT24. Maternal and paternal chromosomes. Yes, you have a chromosome from the mother and father that are identical chromosomes with the same genes in the same loci but different alleles. So homologous pair is from your mum and your dad. That's why one's black, one's red, whatever. And just that they're from different parents, but they can contain the same genes in the same location, which we call loci, the answer to this one here, or locus, loci plural, locus singular, the same location, but they have different alleles. So different versions. You could have brown eye, blue eye, you get the idea, but every human has the same on their same chromosomes. Right, what are we looking at? mRNA, tRNA, can't be bothered. This is a tRNA molecule. By the way, a lot of people don't know what W is, so just to remind you, what is W on a tRNA molecule? I know you, man, know what X is. What is X, peeps? All right, Desi Pink, a sister chromatid is an identical copy of a chromosome. So it's an identical copy. So that's different. When you have your homologous pairs during meiosis, you form you have a homologous pair, and you have sister chromatids in mitosis as well. But in mitosis, you don't have to line up in like homologous pairs or whatever. So in sister chromatids are basically two identicals. So let me show you. Let me show you quickly over here while people answer what W and X are. So if I was to do, this is from Daddy. This is Daddy's chromosome 17, right? That's Daddy's chromosome 17. We're going to join during the interphase, the S phase. We make two copies, identical. Daddy's 17 and Daddy's 17, right? They are sister chromatids. They're identical. The location of the alleles is the same, obviously. That's the same on any number 17 chromosomes. But even more than that, the actual the alleles are identical. So these are two identical. That is sister chromatids. That's different from a homologous pair. So a homologous pair would be this. The homologous pair is mum's 17 with two sister chromatids which are identical. Alleles are identical. Now mum's alleles are not identical, but this is now a homologous pair, and each one is made up of two sister chromatids and held together by a centromere. Then hopefully, during meiosis we get something in the prophase of meiosis 1 where you get swapping over, and these wrap around each other and form a chiasmata or a bivalent, and they swap alleles. So we get some variation there in the prophase of meiosis 1, and that is something that can come up. W and X, people, who got W? W's the hard one. X ain't so bad. So X is anticodon. Yes, the X is the anticodon complementary to the codon on mRNA. Boom! But W is the harder bit. Oh, very good, Jessica. Yeah, it's the amino acid binding site, or NDI 2727. Amino acid binding site. So if you don't know that, that is where each tRNA can specifically like bind to a specific amino acid. So for instance, this one, I don't know what CUC is, by the way. I can't bother to look it up. I don't know those off by heart. I'm not that kind of nerdy. Well, I wish I was, but I just don't recall all that. So it could be glycine, or, you know, it could be cytosine, whatever. Oh, that's a nitrogenous base, so it wouldn't be cytosine, would it? I don't know. Arginine. So basically, it could be different amino acids. Remember, there are 20 amino acids found naturally occurring amino acids, and so you're going to have 20 different binding sites, at least, specifically different ones. We don't have to know what they are, just that they're binding sites. Okay. Oh, shall I go through? Does anyone want me to go through protein synthesis? Just make sure you know. I will do graph questions. I will do 2018 paper. So hard. What example? Non-disjunction is where the centromere doesn't split. Yeah, non-disjunction happens during meiosis. You don't really have to specifically say the centromere doesn't split. You can just say there are problems, with the separation of chromosomes during the, prophase and anaphase. It's going to be during that. They don't separate completely. So, and that's non-disjunction. And that ends up with Down syndrome, extra allele, extra, you end up with an extra chromosome. So if you end up with like 26, 27, I can't remember how much a human has. What do we have? 23 pairs. 46. You end up with like 47 chromosomes. Right, that's right. Non-disjunction. Right, does anyone want me to go through protein synthesis, transcription, translation, or just loud at? What are you saying, peoples? Anyone want me to go through and I'll whiz through quickly, or? All right. No way, Imran Gaming, the hardest thing ever. Yes, no. So we got kind of a mix. I'll speed through it, right? So we just want to go with quick. I'm just going to speed through it. Quick 5 markers. So we're in the nucleus. What do we need to know? Here is your DNA. Your DNA is going to open at the specific site of a gene. So unlike DNA helicase opening the DNA, unzipping the strands, we are just going to say this. So, yes. This is going to be, that's supposed to be like the universal symbol for an enzyme, right? That's like, this time we're not saying DNA polymerase. This is RNA polymerase. And I would say in transcription, if they don't use the word transcription in the question, you might get 1 mark for the word transcription. All right, it's an A-level term generally. So RNA polymerase, we want to say, joins the DNA kind of at the start of a gene. At the start codon. Remember there are, what is it? What do you call it? What is it? The degenerate nature of DNA. There are more than one amino. Blah blah blah. We'll talk about that in a bit. There are more bases. There are more codons than there are amino acids. 20 amino acids, and most of the amino acids have more than one code. But we have start codons and stop codons, like AUG or whatever it is. So RNA polymerase joins the DNA at the start of a gene. So it's going to find that start codon and it can bind there. And then if we want, we can say DNA or whatever. We might want to say H bonds unzip. Or a broken whatever. And then we want to say, here's where it gets a little bit difficult depending on whether you're OCR or AQA. If you're AQA, they're often a little bit less technical, slightly less technical. So we can just say free nucleotides. Now, you should really call them RNA nucleotides. But often I've noticed on the AQA spec, they just say free nucleotides. So free RNA nucleotides join by, and I can't be bothered to write complementary, so comp base pairing. Make sure you're thinking I'm A-level. When you want to get tempted to just drop words, you're A-level, remember, so you need to point out that. So free RNA nucleotides joined by complementary base pairing. So what does that mean? So the key differences here between semi-conservative replication of DNA and protein synthesis is that we have RNA nucleotides, not DNA nucleotides. They're free. And we have RNA polymerase, and the whole strand doesn't unwind, just the strand at the gene. I tried to say that there, but I messed it up. H bond unzips. What the hell does that mean? You should say the hydrogen bonds unzip for that gene. Then what's going to happen is these free RNA nucleotides joined by complementary base pairing. This is our chance to show off our knowledge and say G to C, but this time it's uracil, isn't it? Instead of thymine, it's uracil. And uracil to adenine. Do I need to write uracil and adenine? Not necessarily. I could have put U to A there. But you see what I'm saying? So I'm pointing out that the free RNA nucleotides, the difference is they have a U instead of a T. So it's U joining on here. Right. Then we just want to say, we want to just do the same thing. We want to say the polymerase enzyme. If you're in the exam board that is explicit, like OCR, you've got to say RNA polymerase. Moves down, forms, same thing as in replication, forms phosphodiester bonds. If you want condensation reaction, whatever, forms the phosphodiester bonds by condensation reaction, blah, 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 blah. I'm going to keep it short. Once stop codon reached, once we reach the stop codon, the mRNA detaches. I don't know how to spell detach. I'm going to be honest. The mRNA detaches, leaves via a nuclear pore. Now, if it's 5 marks, I've definitely got my 5 marks, right? I might have missed out some technicalities there, but it leaves via the nuclear pore. All right. Now, that is the most messy, disgusting, horrendous thing. Yes, transcription is the production of mRNA, then translation is for forming the polypeptide. Absolutely. Just right, hydrogen bonds break, yeah. Protein synthesis is transcription and translation. It's both. Protein synthesis involves both. So we're going to do both. And so what's going to happen next is we're going to have a ribosome. That is the worst ribosome in the world, okay? So we're going to have our ribosome and our mRNA. They'll be specific and they'll tell you if they don't want you to talk about transcription. And then we're going to have translation. So part of protein synthesis is the transcription to make the mRNA, which is going to carry the code out of the nucleus. Of course, it's going to be spliced and introns are removed. So you won't necessarily have to go into that detail, but introns are removed by splicing here. And then our form of the mRNA, our spliced form. So we have pre-mRNA and then we have mRNA, which has been spliced and had the introns removed. That then comes here, and all we're going to do here is, this is the codon, and then we're just going to say down comes, remember we just looked at a tRNA, so we just want to say down comes our tRNA, bringing with it an amino acid, and we want to mention anticodon and codon joined by complementary base pairing. So I would just say, yeah, like tRNA brings amino acid. Oh my days. Running out of room. tRNA brings an amino acid. Say where the codon is. Codon on mRNA binds to anticodon. And say where it is, just in case. They get funky, don't they? On tRNA. And a chance to say by comp base pairing. Okay, so that's going to get you some marks. Then what we want to say is a second tRNA. So this is my first tRNA. A second tRNA is going to come down and bind. Now imagine these are bound down here, and only when the second tRNA binds, brings another amino acid. So I'm not going to repeat all of that again because that's long, but second tRNA brings a second amino acid, and this is when you talk about, you can say binds by complementary base pairing. You won't get marked twice for that. We then want to mention that peptide bond forms between the two amino acids. So we want to mention peptide bonds form between amino acids. Oh my days. So we want to mention that peptide bonds form between the amino acids, and then the first tRNA, I've run out of room guys, so then the first tRNA leaves, and then a third tRNA comes, bonds, peptide bonds, complementary base pairing, hydrogen bond to the mRNA with the tRNA, and then a peptide bond. So you now have 3 amino acids. The second tRNA leaves. So there's always one tRNA in contact, keeping the protein, the polypeptide chain that's building, attached. Then a fourth tRNA comes. The third one can bugger off as a peptide bond forms. So they're all connected. Eventually they reach a stop codon on the mRNA, and we can say the polypeptide detaches. And that's when, if this is attached to the rough endoplasmic reticulum, folks, the polypeptide will go inside the rough endoplasmic reticulum, where it gains its secondary structure. You know, it's going to be able to form a secondary structure. Just remember to remember the protein structures. So the order of the amino acids was decided by the mRNA, was decided by our DNA. So the order of the amino acids is known as the primary protein structure, and that's really important because that informs the final 3D shape. Now, the order of the amino acids, the primary structure, tells you the eventual 3D shape, but it only has peptide bonds between it. So in the primary structure, the only bonding we had is peptide bonds. Literally seconds after this polypeptide is made, it will begin to fold, forming hydrogen bonds, and it will all either be a beta pleated sheet or an alpha coil. They're the two different secondary structures you can have. A beta pleated sheet is very like that, like a sheet of paper that you've folded into squares or rectangles and then opened up again, whereas a coil is like a spring like that, you know. Anyway, so it can either be the alpha coil or the beta pleated sheet. Now that has obviously still got peptide bonds from the primary structure, but now has hydrogen bonding as well. And then they will gain their tertiary structure, not because of their beta pleated or the alpha coil. That's not linked to the tertiary structure. It's the primary structure. The order of the amino acids decides which R groups are next to each other. Now, the R groups can either form ionic bonds, or hydrogen bond between each other, hydrophobic, hydrophilic interactions, and because of that, you get further folding to form the 3D shape. Now, normally, a single polypeptide chain only has 3 levels of structure, and the final 3D shape is decided by, it's called the tertiary structure, and that's decided by the primary structure. However, if you are like hemoglobin, which is made of more than one polypeptide, it has 4 chains, or say an antibody, which has 4 chains as well, 2 light chains, 2 heavy chains, so more than one polypeptide, then your final 3D shape is called your quaternary structure, which is how all the different polypeptides interact. Anywho, that was just a quick rundown of the old protein synthesis. Quick multiple choice. Which row, A to D, shows the correct codons and anticodons? I'll get off of this now. All right, let me go on to some mass transport then. So, I'll just quickly go over the answer for this. If anyone knows the answer, put up the answer. Anyone know the answer for this? And then I'll move on. We'll get off of this. Secondary structure of the protein is either an alpha or beta sheet, Jessica. and it is formed by the different hydrogen bonding. Okay, so original is CCG, the mRNA GGC. That's correct. That's correct. The original is TTA. That is going to be AAT. Can you have T in mRNA? You can't. So that row is buggered. CAT, GUA, that's all good. UUA, that's all good. That's all good. And then you've got to check, bloody hell, this is long, I'm going to kill myself doing this. And then I've got to check this off against that. GUA would be C. U is basically a T, so that is correct. I think it's going to be B, you know. AAU, that would be, those are basically like T's, so these would be A's, that's correct. That would go with it, yeah. And then U to A, and then C and G. Oh my gosh, it's B. I'm going B. It's B. Yes, well done. Boom. Okay, right, we're going to move on. I'm probably going to do a bit of mass transport, guys. I know people are like, sir, can you do some evaluation, application questions? I'll tell you what, we'll do some evaluation questions. Boom. We'll do some evaluation questions. I'll have to look up and find some application questions. Which is going to take me time, peeps. So let me just. Right. Why do some mutations not cause a change in the structure of a polypeptide? I'm going to go on to some evaluation questions next. Some of the 6-mark evaluations. So we'll have a look at some of them. Graph and 6 markers. I have got mass transport still to go through and a bit of like Simpson's index and all that business. And then heart and all of that. Where are we? Nice one, Natra. Yeah, the mutation could occur in introns. So if it doesn't occur in an exon, it's not going to change a polypeptide, is it? So well done. It could occur in an intron. And of course, we can get a silent mutation. Do you all know what a silent mutation is? And that's where, because DNA is degenerate, and there is more than one triplet code for each amino acid, you know, you might get GGC might code for the same thing as GCC. So if the middle G turns to, you get the idea. They both may code for the same amino acid, so it doesn't cause a change in a polypeptide. So there's our 2 reasons. Yes, the codes are degenerate, but you need to explain that so that the mutation will be silent if the new code, the mutated new code, still codes for the same amino acid, then it will be silent. But also the mutation could occur in non-coding DNA. Excellent. Let's move on. People want to do, I'm going to do some evaluation questions. So let's have a little look at the eval questions. All right, let me see if I can get this up without it all going Pete Tong. Oh, and it's gone Pete Tong. It's gone Pete Tong. It has gone Pete Tong. It has gone Pete Tong. Not there. I want to go here. Okay, what have we got, Bob? It has gone Pete Tong. There we go. Oh yeah. Woo! 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 This looks fun. Yeah, baby. Oh, rather stick pins in my head. Yeah, what can I say? Don't worry, not long till the exams.
I can't handle the stress no more, dog. These damn A levels.
Okay, let's have a butcher's hook. Listen, you got to know about exam technique though, haven't you? You got to know how to speed it. You got to know how to speed it. Haven't you got evaluation coming up tomorrow? Sure, you're going to have some evaluation questions tomorrow. It's not just paper free. You got to just know how to speed it. Look, use the information to evaluate the use of BSCT to treat HIV infections. So all we want to do is go through and see, have they done anything good? And there's always at least 4 crap things they've done. So current treatment for HIV involves the use of daily antiretrovirals, ART, to stop the virus being replicated. Only 59% of HIV positive individuals have access to ART. So, okay. So not many people have access to ART. So that means scientists have found that 2 HIV positive patients, P and Q, straight away have got into remission and have no detectable. Straight away, two people. Hello, mate. Is that good? Only having 2 people? Whoa, big scientists. They should be doing 1,000 people, but still, 2 HIV positive patients, P and Q, have gone into remission and have no detectable HIV. This happened after a blood stem cell transplant called BSCT. So they had a blood cell, whatever, stem cell transplant, BSCT. They've gone into remission. They have no detectable HIV. Again, evaluation. Just because they can't detect it, they could still have it. So we've got to be thinking, evaluate. It's a language game. Patient P was given 2 BSCTs and patient Q was given 1. Does that make it a fair thing? No, but at least we can say you only have to give someone 1 BSCT and it seems like it goes into remission. So that'd be on my positive. And then all BSCTs came from a donor with T helper cells without the CCR5 receptor. In addition, patient P had radiotherapy. Oh my God. So we don't know if patient P was it the radiotherapy or was it the BSCT? So we've got loads of things to rip this evaluation apart. And patient Q had chemotherapy. Oh my days. So they weren't really controlled very well. Patient Q, maybe it's the chemotherapy that's got rid of their HIV. Both treatments are toxic. So the other bad thing is, these are toxic treatments, which means we don't know if the BSCT or these are causing damage. It's a load of bad things with this. Both patients stopped receiving ART 16 months after BSCT. 18 months after stopping ART, both patients had no HIV RNA in their plasma, no HIV DNA in their T helper cells, and no CCR5 on their T helper cells. Okay, so basically, you just got to rip this one apart. There's some positives to say. Yeah, and then, listen, I know sometimes it says ignore. It's going to say ignore 50-50, but ignore doesn't mean you lose a mark. We'll point out that it was a small sample size and there were no stats tests to see, well, there isn't really anything to do stats on with this, but you might as well jam it in there. Get in the habit of jamming in no statistical test to see if the difference was significant or something like that. Get in a habit of jamming that in there. 50% of the time, it will be on the mark scheme. You can kind of tell when it is or it isn't. Here's the answers, folks. Look, there's so many things. So, you're not, yeah. So this is these are the good things. You probably want to get like at least one of those, and then there's absolute crap loads of against. We don't know which treatment is having the effect because they had chemotherapy, radiotherapy, and BSCT. It might be the chemotherapy or radiotherapy. They only did it, by the way. If we have a look here, this is the other thing that I missed this is the thing I missed, which is in every single evaluation question, the statement they want you to evaluate says HIV infections, but they did HIV-1, so you have to have eagle eyes. So just because they found out that this treatment was good for HIV-1, it doesn't mean it's good for HIV-2, 3, whatever the other types of variants of HIV are. So this only works for HIV-1. We don't know. We don't know if it'll work in all people. They only did it on 2 people, so they haven't done it on women, men, young, old, fat, thin, you know, different ethnicities, whatever. Only so there's loads and loads of stuff. We don't know after 18 months. We don't know long-term effects. Anyway, you get the idea. I just want to show you that let me just show you that in these evaluations, this is definitely one with a stats test. How do I know this one will lend itself to no stats test? Because look, they're comparing 2 groups. You want those error bars overlap, don't we, to see if there's a significant difference between group T and group C. Even if I haven't read all of this, I know straight away it's going to say evaluate the student's conclusion. Let's not even read the information. Let's pick up some marks without doing any work. A student concluded from figure 4 and 5 that training has a positive effect on VO2 max and CS activity. Well, evaluate the student's conclusion. What kind of thing did they do? They did an 8-week training program. The following measurements were taken at the start and again at the end. VO2 max, measure of maximal uptake. Citrate synthase activity is an enzyme involved in the Krebs cycle. The scientists then calculate the percentage increase in each measurement in both groups, shows their results. Well, we don't know. Percentage increase in the VO2 max, percentage increase before and after. So we can see that group T, it had a bigger effect on group T, the VO2 max, than group C. Yeah, both. So again, evaluate. We can say, look, VO2 max and CS activity have both had a positive effect in group T and group C. However, right, so we can say there has been a positive effect. It's had a bigger effect on group T in both VO2 max and the CS activity, bigger for group T in both cases. It still had a positive effect on group C in both cases. It's increased their CS activity and increased their VO2 max. So yes, we can agree with him there. However, straight away, look, they've got none of these things. They've got none of the error bars where you can say, oh, there's no we don't know if there is a significant difference between T and C, and we don't know if this percentage increase is a significant one compared to their before. So we don't know if these differences are significant because they've not done a stats test between group T and C, and we don't know if the percentage increase is significant because there's no stats test. So coolio. And there's probably other things as well. We don't know the size of the group. So if I was evaluating it, we've not been given information about the group. It was only an 8-week training program, etc., etc. So look, the positive is that it went better for both groups, but no statistical tests, so we don't know if differences are significant. No statistics. We don't know if it's down to chance. Only 8 weeks training. Training didn't last long. Might not be true for all exercises, regimes, and oh, I didn't even spot this was only men, was it? Oh, it didn't say. Did it say males? Ah, well, maybe somewhere. I didn't see that. So you get the idea, right, peeps? Oh gosh, why the hell are adverts popping up on my thing? Alright guys, very similar. Very similar. So sports scientists investigated whether there was a correlation between the percentage change in VO2 max and the percentage change in C activity in group T. Okay. percentage VO2 max, percentage change in CS activity. Okay. Having thymine at nucleotide position 16,519 in group T causes an increase in ability to exercise longer. Evaluate this conclusion. Use all the data. So, what the arse? So these are from the previous one. Well, how do we know any of that? I don't know whether I've disincluded stuff, but oh, yeah, whatever. I don't know if I included it. No. What have we got? We got anything else? Got anything else? Look, there's another one. 5 marks. I can't be bothered to do that. It's going to be the same kind of thing. So it's too long to do evaluation questions. Let's have a look. I'm happy to have a look at a paper, but I think I'm going to go through a bit of mass transport. Once you've done one evaluation, it's all the same. So like you're going to look here and look, just spot the they do the same thing most of the time. So when you're in your exam and you get this dirty question, don't be scared of it. Look at this question. Using all the information, evaluate the effectiveness, check this, of using myocausi to increase crop production under conditions of water shortage and no water shortage. I'm guessing already, we haven't looked at this yet, that they didn't do theirs on myocausi, or they did it on a specific type of myocausi, not all myocausi, which is a fungi. And it says crop production. Are they going to have done it on all the crops in the world? Wheat, carrots, potatoes, whatever, yeah, peas, rice. No. So they're going to have used the specific crop. Oh, look, have a look, folks. They've done it on tomatoes. So straight away, spot in the sentence, you want to, rather than have to read all that crap, they've said crop production. They've gone and done their experiment on tomatoes only, and then Brains here has made, oh yeah, it's great, you know, on crops. Well, they should have done it on other things. Now let's see what type of species. Scientists investigated the effect of the myocausial species Glomus intradices. Okay, so any positive effects are only for that species. So there's a second mark there out of 5, and we haven't read the information. So there's always tricks in the evaluation sentence that are going to smash you marks. Now look, they have done statistical tests here, but this gives us stuff to talk about, whether it was significant or not. So look, when there was a water shortage, have a look, and I haven't even read the information yet, but I can say there is a significant difference between Q and R, Q and P, because these bars, these standard deviation bars, don't overlap. So I'm going to say that there is a significant difference when there's a water shortage between Q and P. What are Q and P? Greenhouse, Q is treated with seedlings with water shortage. Oh, treated. So the treatment works during water shortage. They get a greater mean mass of tomatoes. So that's a positive, right? That is a good thing to talk about. Over here, when there's no water shortage, yes, it looks like S, which has been treated, has the special treatment, and R is like your control without the treatment. But no, there is no significant difference between the mean mass in S and R. So that's a negative. So look, there's loads of negatives, and we haven't even begun to pick this apart. So I'm just trying to tell you, without having to read all of this, that we can see, and then we can look at how many they did. Look, it says after 60 days, the scientists, so they did it for 60 days. You can say good things, like if they did the study over a long time, or they did like millions, or they had thousands of people in their sample, or whatever, you can say, listen, that's good. The other thing, look, where did they do this stuff? Did they do this? Okay, where do you grow crops? In a field, right? Crops are grown in a field. Did they do this experiment in a field? Let's read. Scientists planted tomato seeds in a large number of pots containing sterilized soil Is the soil sterilized in a field? They added a culture of G intradisease to 50% of the pots and 50% were left untreated. After the seeds are developed into seedlings, young plants, the seedlings, untreated and treatables, were planted into 4 separate large greenhouses. Okay, maybe we can say they're in a greenhouse. So again, this is not in an open field. A very limited supply of water, blah, blah, blah. After 60 days, the scientists determined the mean mass. So it lasted 60 days. Let's have a look at the answers anyway. But I'm just trying to shorthand. There are ways that you can pick up marks on these evaluation questions without having to cry into the exam paper, okay? There are standard exam techniques like dissect the sentence and you will find 2 or 3 marks in the sentence. So look, these are the answers. You can pause and have a read through these if you really want to have a look, but you can see there's no overlap, no significant difference. There's a significant difference between S and Q, but then with R and S there isn't. Only one mycorrhizae species, only for tomatoes, not for other crops like rice, potatoes. They did have a large sample size, so they're saying that that was good. They used a large sample size. It was done in sterile soil and in a greenhouse, not done in a field where you would grow crops. Again, I just want to point this out. I'm not going to go through this whole one. Look, I'm not going to go through all of this. It's too long and you ain't got that kind of time before your exam. But I just want to show you. Let's read that key sentence in an evaluation. These are all AQA key evaluations that have come over the last 6 years. So look at the technique. That the student concluded this investigation showed that ammonia, let's just make sure that we're using ammonia. We might be. I haven't looked at it yet. Inhibits nitrogenase activity in nitrogen fixing bacteria. Evaluate. Let's first of all, have a look. They're using nitrogen fixing bacteria, but all right, let's see what they did. Nitrogenase to produce ammonia from nitrogen in the air. A. Crococcum. A. Crococcum can use ammonium chloride as a direct source of ammonia when a source of ammonia is not available. This bacterium uses nitrogen fixation. So yes, they did do nitrogen fixing bacteria, but once again, peeps, they did Azobacter crococcum. That's a nice word, isn't it? Crococcum. Yeah, so again, that's a problem, isn't it? They've said all nitrogen fixing bacteria. They've implied for all nitrogen fixing bacteria. They've only done that. I can't be bothered to look at anything else. Same volume, so that's good. They did control variables. Different all right, they grew it in ammonium chloride rather than directly in ammonia. Yeah, they've done ammonium chloride rather than ammonia. It can use ammonium chloride as a direct source of ammonia, but it isn't ammonia. So they should have said that they haven't used ammonia, they've used ammonium chloride. So that's a point as well. Look, they used NH4Cl. They did it to get the NH3, but it's not quite the same. And then you would just look, they've done lots of concentrations. That's a good thing, blah, blah, blah. We haven't done stats test and all that business. And that's the kind of answers they were looking for this one. It was only a 4 marker. But look, they only used one species. Ammonium chloride could have had an issue with it being ammonium chloride and stuff like that. Anyway, I didn't bother going through all of that. I just want to show you the common ways to pick up the marks. So here's another one. I'm not going to go over it massively. Just look at this. Group B, rats could be used for studying type 2 diabetes in humans. Use the information to evaluate this conclusion. Rats should be used. Let's have a look. Right, they used laboratory rats. So straight away there's a difference. This did not say lab rats. This just said rats. Laboratory rats are different than rats that live in the wild. So we've got a problem there. With the characteristics of type 2, blah, blah, blah. So you get the idea, and then there's loads of other crap going on here. And when they've done the statistics test for you, that's great. You get to see whether there's significant difference and talk about it. It's just easy marks. So look, the mean body mass between group A and B, does it overlap? Well, how would you know? Well, go 221 plus 3.28 equals whatever the hell that equals 3. Oh, Sugar Ray Leonard. 2, 4. Oh, Fudger Lubber. 2, 2. I ain't got that kind of time. 2, 2. 224.28. So look, no, there's no over, and then you do 233.34 minus that and see if there's any overlap. Anyway, basically add that to that, minus that from that, and see, do those numbers overlap? If there's no overlap, we can say there was a significant difference in the mean body mass between A and B. So use the plus and the minus, see if there's overlap. It's a bit harder when you've done that. You've got to do some calculations, but you can say that's 2 marks out of 5 there. One for saying, is there a significant difference in the mean body mass? Is there a significant difference in the glucose conc? I imagine there'll be a significant difference in one and there won't be in the other. And yeah, so on your evaluations, you should be able to get some marks easy peasy like that. It goes on and on and on. We will get off of evaluations, people, but there is the answers for that one. Oh my gosh, look, SDs do not overlap for blood glucose, do not overlap for mass. So there was a significant difference in mass and there was a significant difference in the glucose concentration. So that would have picked you up 2 fat marks. And then investigation was done on rats, not humans. I didn't even spot that they were implying it about humans. I didn't get into it enough. Long-term effects there, blah, blah, blah. Anyway, let's move off of this. I'm going to go through, I think, a little bit. Let me bash through some. I want to just go through some. I might do a bit of. Let me go through a little bit on one of your topics that's coming up, which is transporting plants. And then I'll go through transporting fish gills and just remind you of that stuff. Oh my days. Oh sugar, what am I doing? I'm clicking on everything. It's all going techno fear. Techno troubles. That's right, Paige. You've got plus and minus the standard deviation from the values and see if there's a significant difference. If there is, and see if they overlap. If they overlap, there's no significant difference. Even though the means are different, it's saying, you know, there's no significant difference. If there is no overlap, then there is a significant difference in that factor, and that means that the treatment, whatever it is in the evaluation, had an effect. So there's some easy marks if you get that. So don't be upset. And if they don't have those, if you get a graph and it shows they're comparing 2 groups, if they're comparing 2 groups data and they haven't done statistical tests, then that's 1 mark for saying, look, they should have done a statistical test in order to see if there's a significant difference between the groups. So they either give you the stats and you get the marks for explaining them, or they won't give you the stats and you get a mark for saying, look, they didn't do a stats test, so we can't tell if there's a significant difference. And then there's always a mess up in the species or the application. There's always a disconnect between the sentence, like, this would be a great treatment in diabetes for humans, and then they've done the experiment on rats, or this will be great for like all rats, and they've only used 1 species of rat. So there'll always be a disconnect between the connection as well. There'll be a couple of marks in the actual question, and then you've got the stats, and then, you know, you can evaluate, is it a large sample size, did they do it for a long time, all of that kind of thing, is it representative of the population, all of that business. Right, what am I doing? I just want to whiz through transporting plants because someone put up they wanted to go through transporting plants, so why not, eh? Right, xylem vessel, phloem vessel, vessel elements, dead cells, no end walls, and lignin. Non-lignified regions called pits, which allow water to move out sideways by osmosis. Now, separating the xylem is next to the phloem, and separating them is a region called the cambium. Okay, it's a region called the cambium, which I think has got lots of like stem cells in there and whatnot. But anywho, the cambium is in between, so you can sometimes get a mark for saying water moves by osmosis into the phloem when you do mass transport. The sieve tube elements, so the phloem is made of sieve tube elements with fat companion cells on the side that can do respiration and do active transport and stuff like that. The sieve tube elements have these weird sieve plates with holes in the end. This is a bit of evidence against mass flow in the phloem, the mechanism we know, because some people say you just you can't generate the kind of pressures you'd need to force liquids through these holes. So that's kind of evidence against. There's no lignin in the phloem vessel. It doesn't offer support, whereas the xylem has lignin, so offers support. Right, let's just have a little look. I'm just going to go through movement in the xylem. So nice and easy. Here is your xylem. Your water molecules are polar. So what am I doing? They're positive, and this as lone pairs, whatever. It's got 2 lone pairs. I'm drawing 1 because I'm a rebel. Right. Anyway, what do we get in between? We get hydrogen bonding. This is called cohesion. This is called cohesion. Cohesion is hydrogen bonding, okay, between 2 adjacent water molecules, between the delta positive hydrogen and the slightly positive negative oxygen. You can call it delta if you want. In AQA you don't need to use this sign. You don't have to know that sign in AQA. So you can just say the slightly positive hydrogen and the slightly negative oxygen. You get hydrogen bonding between them in adjacent water molecules. This is a covalent bond. Don't make the mistake of saying there is hydrogen bonding in a water molecule. There isn't. There's no hydrogen bonding in a water molecule. It's between water molecules. So check your language. That's called cohesion. An additional, not on your syllabus, but an additional in areas of the wall of the xylem where it has lignin. These, by the way, are called pits or bordered pits. These unlignified or non-lignified areas are called pits. These regions with lignin, the water can bond to the lignin as well, and we call that adhesion. That is on the except if you're asked how water moves through the xylem. You can talk about adhesion to lignin in the walls of the xylem. Now, why is this vital for how water moves up in the transpiration stream? In the leaf, water escapes by transpiration through the stomata. So make sure you point all of those A-level things out. Water escapes by transpiration. Transpiration of H2O creates, you can call it a pull, a negative pressure. Oh my good gosh. What is all of that stuff? That is really tension. Tension is a pulling force created by the negative pressure of water moving by capillary action across the leaf and escaping. It creates a pull. If there was no tension, the water molecules, yeah, woo, they can stick together, but they're not going to go anywhere. Unless you can have a rope, you've got a rope, but the rope won't move unless you pull on it. So it's the transpiration that creates the tension. And then the fact that water molecules stick together in a continuous water column is the cohesion, right? So it's called cohesion tension. Just make sure you can explain that and you'll be sorted on any questions in the movement of water up the xylem. So cohesion is the water molecules sticking to each other because they're polar, or they have a dipole, if you like, positive and negative bit, slightly positive, slightly negative. The oxygen slightly negative, the hydrogen slightly positive. They hydrogen bond to each other. That forms a continuous water column. If water couldn't hydrogen bond, what would happen? It means when we get the transpiration pull, one water molecule would pop out and the rest wouldn't follow. So that's no good. That wouldn't work. The whole system would fail. It would fail. If you break the water column by drilling a hole or by injecting something into the tree that breaks the water column, then the tree is going to die because you've ruined the transpiration stream. So this has to be an unbroken column of water being pulled like that. So that's called cohesion tension. Great. Let's look at the mass flow. Oh, white. I thought it'd be green. Okay. The other bit of transport you need to talk about is in the phloem. Now, depending on whether you're OCR or AQA, if you're AQA, this is easy peasy, double lemon squeezy. So, if you're AQA, you have xylem here, then you have cambium here, whatever. I'm not going to draw it out. You've got the cambium. Then you have bish. Look at this utterly amazing drawing. Incredible. Wow. This is not the whole of the phloem. This is known as the sieve tube, and these are called sieve tube elements. These are called xylem vessels, xylem whatever, and then you have companion cell. Okay, now in AQA you don't have to know how we get sucrose in here. If you do OCR, you need to know, just for those OCR people, you need to know that we actively transport hydrogen ions out. It's very much like chemiosmosis in photosynthesis and in respiration. It's almost exactly the same, almost, but not the same. You transport out the hydrogen ions, and then they move through a co-transport protein. It's a bit similar to how glucose is absorbed in the small intestine in AQA. Hydrogen ions are pumped out, which is probably why they don't do it in AQA, because they do the same mechanism there. So hydrogen ions are pumped out of the companion cell to create a concentration gradient. The hydrogen ions then diffuse by facilitated diffusion, and they drag sucrose molecules they drag the sucrose molecules with them back in against the concentration gradient. Now in AQA, you ain't got to know all that business, so we're going to allow that, and we're going to rub that out, and we're just going to call it active loading. So in AQA, simplify. You want to say step 1, active loading of sucrose, Bosch, into sieve tube. This is for translocation, by the way, is what I'm waffling about. So sucrose actively loaded into there, same mechanism, hydrogen ions pumped out, diffused by blah blah blah, whatever. Now what does that do? That does step 2. If you bung a load of sugar into water, and sucrose, although it's a disaccharide, remember it's made of glucose and fructose. Remember your disaccharides. Maltose is 2-alpha glucose. You got sucrose is fructose and dirty fly. Yeah. And what else? Lactose is made of galactose and glucose. So remember your 3 disaccharides. You have to remember them. Remember your biochemical test for sugars as well, guys. It could be linked in one of these questions. So yeah, sucrose is a non-reducing sugar. You'd need to add hydrochloric acid first, and or whatever. You'd have to break the bonds with hydrochloric acid and then you neutralize with sodium hydrogen carbonate, and then do your Benedict's test with sucrose. But with the others like maltose or the monosaccharides, you can just do your normal Benedict's. Right, what am I'm waffling, I'm going off the topic. Right, 1, active load into there. 2, lowers the WP. It lowers the water potential because you've now increased the solute concentration or lowered the water potential at the top of the sieve tube. What happens is 3, this causes water to move across the cambium by osmosis from a high to low water potential, right? So I'm just going to say osmosis from xylem. Obviously, you want to explain that. You're in the exam, you want to show off, you want to be like, water moves from a high water potential to a low water potential in a sieve tube across the cambium, you know, if you want. Partially permeable membrane. I mean, that's getting a bit too excited, but you get the idea. Right, and then number 4, what happens? Well, 4, the this makes a high hydrostatic pressure, or we just say increases pressure. Increase in pressure, it's the water pressure or hydrostatic pressure like you get with tissue fluid. So increase in pressure, hydrostatic pressure, HP source. Yeah, water moves down a pressure gradient. And there we go. That's how translocation happens. So this, I know I've got the xylem written there, but it is translocation. And that is the movement of amino acids and the movement of sucrose. So sugars and amino acids move by translocation around the plant. There is some evidence against it. Some scientists say the sieve plates would be too resistant for water to move like this. The pressures that would have to be delivered are just too much. Now there are some There is some evidence. What's the evidence that we need active loading? Real simple. Add an enzyme inhibitor that prevents active transport. Then you can't do the active loading. Then the whole thing flops, right? So that's where they get the evidence for that. No one's actually seen with their eye active loading. It's all from observational experiments. So, yeah. Other things we can add. We can add radioactive isotopes to the glucose, like by using carbon-14. So carbon-14 is a radioactive isotope. It's basically you're labeling the glucose in sucrose, and then we can trace the glucose moving down the plant by using photographic film. As the radioactive isotope moves down, it will decolorize the film, so we can show the direction it moves. You can do that by like killing various parts of the leaf and the stem and freezing them from many plants, and then you'll kind of get like a freeze frame shot of the substances moving down. There's various other experiments you can do with this. You can do the one where you get the bulging. What's the bulging ring? You get the bulge above, and then you could do the aphids. You take green fly and you get green fly and lick a hole in the top of the stem with a plant with a green fly's little proboscis, its tube, and then when you remove the green fly, you kind of pull it out and its head gets left behind. Pretty gruesome, right? And then the sap will come out at a higher pressure than if you whack the green fly in lower down and then yank it off, ripping its head off. Then the sap will come out not at such a high pressure lower down, which tells us that the pressure is higher above than below. So that's like some of the things you can do. Anyway, any questions about transporting plants? Otherwise I'm going to bugger off and do animal transport. Yeah, absolutely. Oh, well done. Yeah, someone's mentioned the ringing. Bit dodgy, right? That's the one where you get the bulge. I know you guys get excited with words like bulge, but you remove the ring, and the ring contains. What does the ring contain? What does the ring contain? Does it contain the phloem? And then what happens is all the sugars build up at the top, and you get the bulge at the top. Whatever the ring contains, it kind of like mashes it up. I can't remember. Yeah, they removed the bark. Yeah. Yeah, it's because I'm tired, HT24. It's tiredness, honestly. All right, let me go on with the animal. Let's have a look at the heart. Let's have a look at the heart. Let's have a look at the heart. I did do a live on all of this in much more detailed, obviously. I did a live a few weeks back on transporting plants where I went through this in like much more detail and a much better explanation. I went through each of the experiments and that in a much better way, but you know, I ain't going to be able to like do the whole of the A-level course in one day. So, what we're looking at, hold on, I'm on here. Where's my heart drawing? Let's have a butcher's hook. I've got a heart drawing somewhere. No, not there. I'm just flicking, folks. Having a flick, and I'll give you man a quick question on this, and then we'll move on. I'll just give you a quick question on this, then we will move on, peeps. Oh, I don't want you. Don't want you. Want you. Okay. Right. Guys, I'm going to quickly go and grab myself a coffee, see if you can get the 5 points, describe the transport of carbohydrates in plants. You might say, so we're never going to get asked that. This was a question that came up a couple of years ago, AQA, paper 1, A-level, boom. I'll be back. I'm back. I'm back. Hopefully you guys have yeah, be different. It be different. It be different. It be different. Yeah, make sure you're not hate watching in your underpants. Make sure you hit the like.
If you push the button,
Like me.
Would you like it?
Like.
Do it. Do it.
Do it.
Okay, right. So you had plenty of time with this one. Just check and make sure if you had a go at this question that you got all of the marks. Sucrose actively transported into the phloem, or sucrose is co-transported, moved with hydrogen ions into the phloem if you really want. But you literally could just say sucrose actively transported into the top of the phloem by companion cells. So companion cells are involved. So yeah, lovely. This lowers the water potential at the top of the phloem, so water enters from the xylem by osmosis. Don't just say water moves without mentioning osmosis. This creates a high hydrostatic pressure. Notice hydrostatics in brackets. You did not have to use the term hydrostatic pressure. Same kind of pressure that's created by the left ventricle in our lung in transport in animals. Then the water moves down a pressure gradient, moving the molecules, metabolites like amino acids and glucose down to the sink. And then what happens at the sink? Well, you've got respiring cells. They're using up the sucrose or the amino acids to turn them into proteins. They're turning them, or into starch. They're turning them from soluble to insoluble. They no longer have an osmotic effect. What does that do? That raises the water potential at the bottom in the sink, and so water moves back out into the xylem by osmosis again. Yes, right. Now let's have a little look, because we want to do a little bit of the heart, because the biggest topics, often the biggest topics that are asked heavily are the cardiac, the heart, and transport in plants, which we've just covered. So let's have a little look. Look. Have I got a heart somewhere, please? Where is the heart? The heart, the heart. No, that's no good for me, man. Where is the heart? That's maths. I don't want to do the A-level maths questions yet. I don't want to even do them at all, let alone yet. Where's some heart questions? Okay. What is this? Mass flow, more mass flow. A little bit of cardiac cycle, a little bit of that, a little bit of this. What's that called? Oh my god. No, that's not what I want. Huh? No, that's not what I want. What the... Okay, maybe. Whatever. Okay, talking at my backside. Ah! What's wrong with this thing? Oh, this thing is being a brute! It's bruting me. Oh man, what is wrong with this? This thing is so stupid. Alright, let's go with that fudge. Man, it's a fly trying to get in my coffee. What's that all about? I don't know what that's about, but that's brutal. Oh! All right, people. OCRA. Yeah, I've covered a lot of stuff that will be in OCRA. Yeah, I will do heart. But this is some of the other exchange and transport in animals. I can't get my for some reason my heart thing's not coming up with my picture of the heart and everything, so I just have to do some crusty drawing of it, which I don't really want to do. Where is... I've got a crusty picture of a heart. That's transpiration in plants. We don't want that. DNA code. We've done a DNA, AS biology, maths questions, eukaryotic cells. Don't want that. Why can't I get this? Why can't I get this thing up? This thing don't want to come up. Okay, I had it up earlier. It's being a pain in my butt. Oh, you know what? Sometimes you just gotta allow it. If you don't want it, then I ain't gonna fight it, man. Yeah, I'm not gonna bother. That's not having it. All right, let's just stick with this. All right, we're gonna stick with what we got. So, I'll talk about the heart. I'll talk about the heart, but I can't get the image up for some reason. It's not having it. It's being disobedient. So, what do you need to know about fish? This is kind of important for you. If you don't know about the fish business, let me just rinse through a little bit about the fish for you. I hope you guys are good. If you're doing OCRA, there is a slight difference if you're an OCRA person. If you are doing OCRA, you need to know about fish dissections in slightly, well, no, it's about the same for both of you. So remember, you can do a fish dissection. And with a fish, with any dissection, whether it's an insect or a fish, this is for AQA, and OCR, remember to pin it to the dissecting board. That's just easy marks. Use pins, pin it to the board, use a sharp scalpel, cut away from the body for safety and health and all that business. Now with the fish, what you do is you lift the flap of skin here, and we call this the operculum. It's a bony flap of skin, and it covers and protects the fish gills. Now you lift that up, the operculum, and then you're going to say remove, using scissors, remove a gill arch. Now a gill arch will then have gill filaments on it. So if you're doing the dissection, I would just try and remember 3 bullet points. It's not going to be more than 2 or 3 marks, a dissection of a fish. So lifting the operculum, you're showing off that you know the name of the flap of skin that covers the gills. It's called the operculum. You lift that, you use scissors to cut through the bone of the gill arch and pull that out. And then you should be able to see all the gill filaments and lamellae. Now, also pin the fish to a board to avoid cutting yourself, and use a sharp scalpel. Easy peasy. Right, let's have a little look then. So what we're going, explain how the countercurrent flow system allows efficient exchange of oxygen in the fish gas system. Right, identify structures F and G. What is F? What is G? F is a gill filament. F is a gill filament. Anyone know what G is? You got to do a dissection for AQA as well. Close, Osman. It is a gill filament, and then G is the lamellae, secondary lamellae, or just lamellae. Now, if you're doing AQA, we call this in AQA a lamellae, G, and we call F a gill filament. In OCR, because they're a pain in the backside, they call that the gill filament, or, if you like, the primary lamellae, and this is the secondary lamellae. So I would just stick to the AQA one, which is that the gill filament and the lamellae are different. But in OCR, they say gill filament and secondary lamellae are different. But yeah, what are you going to do? That's them, man, getting excited. So gill filament and lamellae, and by the way, you guys, it's not part of the question, but I could say, how are the gills adapted for efficient exchange? Someone put up in the chat, how do gill filaments and lamellae help the gas exchange in fish? How do they help? This is AQA. This is the AQA question. This is an AQA question, so yes, you have to know this. Okay, it's a large surface area. So those of you saying large surface area, well done. If you want to say that the gill filaments have thin walls and that provides a short diffusion distance, that's the same as any exchange surface. Brilliant. But if you're asked how does the gill filament or lamellae aid in the exchange, it's because it provides a large surface area for rapid diffusion of oxygen. Okay, we're getting oxygen here, not glucose or amino acids. We're getting oxygen in, CO2 out, whatever. We're getting oxygen in, right? into the blood from the water that is flowing around this. Not air around this, it's water that's all over these guys. So these create a large surface area. Now let me just explain, how does the gas exchange system, the countercurrent principle, affect fish? Again, this is an AQA question for those of you that are wondering, but for OCR you have to know both. So you've got to know this for both, right? So this is important. This is the distance along the gill filament or gill plate. We can call it the gill plate. Now the gill plate is another word for what in AQA they're saying lamellae. You see G, so the gill plate is the secondary lamellae, which is, the gill plate is the same as saying lamellae, right? That is a gill plate. That's a gill filament or primary lamellae, and that's secondary lamellae, which is also known as gill plate. I know it's a pain. The gill arch is the bone down here that these are all stuck to. This would be called the gill arch. The gill arch has got nothing to do with gas exchange. It is a structural thing that holds these lamellae and filaments in place. The lamellae and filaments create the large surface area. So this is the distance along the gill plate, and this is oxygen concentration right here. Now, what happens? Oh my days! First of all, I'm going to show you if they did not have parallel flow. So if the water came in here, if the water came in full of oxygen here, and the blood came in alongside the water, so if they both come in at the same place, so if my gills are here and the water is going in that direction and the blood is going in that direction, they're going to meet at the same place, and that's fine for the first half of the gill filament, or gill plate. That would be fine. But as the oxygen, why is that giving me an arrow? Why is it being so dumb? Don't give me an arrow, you git. I don't want arrow. So as the water flows along, we have a problem. The oxygen concentration in the water will go down as it gives it to the blood, and the blood will go up, but eventually they are going to reach equilibrium. And this is where 50% of the oxygen is in the blood and 50% is in the water. This is a very inefficient system. This here would be called parallel flow, and it's like application. So you need to know that you will now limit, you can only get 50% of the oxygen from the water into the blood, because at that point diffusion is happening at an equal rate. We want to maximize the concentration gradient across the length of the gill plate for oxygen. Make sure you don't just say the words concentration gradient tomorrow in your exam. That's a GCSE thing. You must say the substance that the concentration gradient is involved, or like water potential, whatever. Don't just say, oh yeah, this makes a steep concentration gradient. Say it makes a steep concentration gradient for oxygen to transfer from the water to the blood. Anywho, so what happens? What this means is that the water enters here. Say this is at 0 millimeters, the start of the gill plate, whereas the blood is entering over here at the end of the gill plate. The blood obviously has no oxygen at this point, and then what's happening is, as this blood is the most highly concentrated blood in the fish, and even then it crosses paths with water that has just flowed in, so you're going to get a diffusion of oxygen going from the water to the blood, and then all along the length of the gill plate, even when the water is at its very lowest, the water has only got 10% oxygen. Look, the water has only got 10% oxygen, but it doesn't matter, because it's at that point, at the end of the gill plate, the blood starts coming in with 0% oxygen. I mean, it won't have 0, but you get the idea. So along the entire length of the gill plate, O2 is diffusing from the water into the blood because they enter at different ends. That's the countercurrent flow mechanism. Here is the mark scheme for this, and you will see it says blood and water flow in opposite direction. Notice the wording. This is AQA. And it will be the same for OCR. The diffusion concentration gradient is maintained along the entire length of the lamellae or the filament. I've called it gill plate. Gill plate, before anyone wets themselves, is another name for the lamellae. Okay, so it's this thing that looks like a plate, which is the lamellae. Right, where are we bussing off to now? I've just hit on the old Lungicus. I can't be bothered with lungs. I hope you guys are good with lungs. Let me do, we don't want to do lungs. Let's not do lungs. I haven't got a heart picture because I have a PowerPoint of the heart and it won't come up for some reason on my it won't come up on my streaming thing. It's not doing the do. Which is really rubbish because what's this flipping thing doing? What the arse going on here? Oh my days. Oh my good gosh. Okay, so anywho, some people said, sir, what about the cardiac cycle? Oh my gosh. I'll go through cardiac cycle, the heart. Okay. I'm going to carry on for a little bit. I'll carry on for a little bit. All right. Oh my God, what's going on? I'm definitely going to finish in about 45 minutes, but let me just zing the heart. I haven't got anywhere near. I never get through what I want to get through because I have a habit of waffling. And now look, I'm having to draw the heart. This is just painful, people. This is painful. I don't really want to do this. Really doesn't look like this, but never mind. This is painful. I don't want to draw the heart because this is some. But what am I going to do, hey? What are you going to do? Needs must and all that. Right, so what we have here... be quiet, phone. I know it's 8 o'clock. You don't need to tell me, mate. Right, what we have here, don't forget, this is the try before you buy, the tricuspid. But guess what? You just call this AV. If you don't already know that, just call this the AV valves. You want to call it AV valves. That stands for atrioventricular. So between the atrium and the ventricle. Now, cardiac cycle. So let's show you the cardiac. Let's show you the cardiac cycle. They write it in terms of one. This is the way they write it. I would start with 3, but I'm not going to start with 3. I'm going to start with 1. Which is how they write it in all the textbooks. So I'm going to start with the textbook. Personally, I'd start with 3. It's the cycle, and that to me, 3 is the sensible place to start. So I'm going to start with atrial systole. Atrial systole. What does that mean? It's not pasta. It is where the atria contract. Now, I am not going through the whole thing, right? What do you think will happen if the atria, my right atrium here, what is going to happen when that contracts? Well, what's going to happen to the volume and the pressure? Well, the volume will decrease, the pressure will increase, forcing the blood into our right ventricle. Ta-da! Okay, and then you have your pulmonary artery over here. Oh my good gosh. Goody good gosh. So this is our PA. This is our pulmonary artery. And so, of course, deoxygenated blood is coming in here, guys, from the vena cava somewhere back there. I know it splits into 2 and all that, and I haven't done that. There's some crusty drawing. But yeah, that goes into there. Now, I want to come to 2. 2 is going to be ventricular. I will talk about the control of this as well. 2 is ventricular systole. And ventricular systole, guess what happens here? So obviously during atrial systole, the pressure is going to get high in there. And these valves are going to be, the blood is going to be forced through these AV valves. Now, when the ventricle contracts on the right and the left side at the same time, so it's going to be number 1 there, number 1 there, number 2 here, and it's going to be number 2 over here as well in this one. When the ventricle contracts, of course, what is going to happen? Well, what is going to happen is the volume is going to get smaller, the pressure is going to go up, and the blood is going to be forced up through the semi-lunar valves. So you've got these valves somewhere up here in the pulmonary artery. These are semi-lunar valves. Now, they're going to be shooting up there, right? And that's the oxygenated blood. So again, you're talking about how does this apply to grafts and stuff? Well, obviously, if the ventricle's contracting and we're in ventricular systole, I'm going to see a high pressure in the ventricle. They normally do the left atrium, left ventricle, the aorta. That's the graph they normally show the pressures and volumes of. But you would expect the pressure at the start of ventricular systole, the pressure in the ventricle is going to be high, and then the pressure in the blood vessel is going to be high after that, and the pressure here is going to be higher, but this is going to generate the biggest pressure, and then you're going to have the pressure there, right? Now, these are semi-lunar valves. Remember that these shut, the shape of these tricuspid, or the AV valves, just call them AV valves, forget tricuspid. The shape of these AV valves is such that when the pressure of blood below them is great, they shut. The shape of these is opposite shape to these, so that when the pressure below these is bigger, these open. And it's to do with their 3D shape. That's why these will open when the pressure below them is big. We want these to shut when the pressure above them is big. We want these to open when the pressure above them is big. So it's just obvious. You would expect when the pressure is really big here, we would expect these to be closed to prevent backflow. When the pressure is really big here, we would expect these valves to be closed to prevent backflow. So when you have big pressure below a valve, or in this case above a valve, you'd expect those valves to be closed. Anywho, what am I waffling about? We're then going to have, by the way, at the same time, oh my good gosh, shouting into the mic. At the same time, you got the, oh my god, what the fudge is that? You got the left atrium. This is number 1 as well. That is atrial systole. They happen at the same time, double circulatory, so left and right contracting at the same time, blood coming down here, and then, oh, this is going to get messy. How am I going to do this? I've kind of lost the plot of how to do this. What is that going to be? We'll just run with it like that. We'll go with it like that, and then like that, and then we'll just draw some valves there. This is the aorta, remember? And don't forget, the aorta, that is your left atrium. This is the pulmonary vein. And then the aorta comes up, and then it zings off down the bottom behind the heart and shoots off that way. So anyway, yeah, so the oxygenated blood goes in there, goes in here, and this is your LV. So that is number 2 as well. So during ventricular systole, both ventricles are contracting. Most of the graphs are on the left side because you get much bigger pressure in the left ventricle and stuff, and in the aorta the pressure's bigger. Okay, what else? Then we have the final bit is just called diastole, and it is during diastole, both atria and ventricles are relaxed. Now, what's a little bit weird is that during the relaxation, you've still got blood trickling in, and these are open, so the blood can trickle in and fall by gravity into the right and the left ventricle. We've got no pumping going on, it's just gravitational pull. So you do have a trickle of blood going in there while all the muscles are relaxing. Okay, anyway, that is that. Okay, someone mentioned smooth and muscular walls in the artery. Smooth walls in the artery are muscular. They are made of muscle. If you're talking about elastic and muscle, so in your arteries, the walls are made of elastic fibers and muscle fibers. The muscle fibers are for smoothing the flow of blood. That's the best way to say it. They can contract and relax. That's all a muscle can do. So if you're writing about muscles, they can contract and relax. That's all they can do. But that smooths the flow of blood. The elastic fibers help with something called recoil. Okay, they basically, when the artery and the blood goes through and it springs, they help with the springiness. They allow recoil, the elastic. Otherwise, it'd be like a metal pipe and it would just crack and snap because you've got a lot of movement, right? So they allow with recoil. Now, the type of muscle that is in your artery, here's where we don't want to get confused. The type of muscle that is in your artery and in the lumen of your intestines as well, so your small intestine, your ileum and all that, is called smooth muscle. And smooth muscle is involuntary, so you don't have to think, contract my arterial muscle or my artery muscle to smooth the flow of blood. That's happening without you thinking about it. It is an involuntary muscle, so it contracts without you thinking about it. Okay, whereas your skeletal muscle, you contract. And then you have a third type of muscle, which is cardiac muscle, and that is slightly different because cardiac muscle is myogenic, which means it generates its own electrical impulse. Now if I rub this off, I might as well... Is control of heart in paper 1 as well? I can't even remember if control of the heartbeat. No, control of the heart is in paper 2, right? Elastic fibers stretch, not elastic muscle. Yeah, smooth muscle reduces friction. Yeah, but don't talk about elastic muscle. There's no elastic muscle. And yeah, the muscular walls can withstand the high pressure. Smooth muscle walls contract. All right, I ain't going to go over that then. I'm not going to go over the whole SA, AVN and all of that business then. Oh, you got your B, O, H with an R. We got your bore shift. Boring. Use the information in the graph to explain how the seal's myoglobin dissociation curve shows the seal is adapted for diving. Well, seals are diving mammals. They fill their lungs with air. Okay, so let me just allow this, and I just want to actually explain this. So forget this question. I'm going to use this graph to explain. Oh, I don't like the shape of that curve, but never mind. I'm going to use this graph to explain. Now, this could be adult. Forget the question. I'm not referring to the question. This could be adult hemoglobin, HB. And this could be fetal hemoglobin. They love the old fetal hemoglobin and the adult hemoglobin, right? This oxygen saturation, this says blood oxygen concentration. On lots of these graphs, it will say oxygen saturation. Think of this as affinity for oxygen. Think of this as environment. What do I mean by that? In a high partial pressure, we will be dealing with the lungs. In a low partial pressure, we are talking about the muscles, right? So when you answer these questions, the mark scheme wants you to mention partial pressure of oxygen and oxygen saturation, but it needs you to talk about them in terms of environment and affinity. So what we would say, if I was comparing the fetal hemoglobin to the adult hemoglobin, I would say adult hemoglobin, or if I talk about the fetus, I say the fetal hemoglobin has a higher affinity. Look, affinity. So yes, you could say it has a higher saturation of oxygen. That means that it has a higher affinity for oxygen at lower partial pressures. So as the partial pressure gets lower, the fetal hemoglobin still has a higher affinity for oxygen than the adult hemoglobin. Okay. Why? Because fetus is in a low oxygen environment, generally occurs in a low oxygen environment. So their Bohr curve is shifted, well, their curve, S curve, is shifted to the left. Now, if I was just talking about, oh my good gosh, if I was just talking about an adult's hemoglobin, let's pretend this is not a seal, this is adult hemoglobin, then how would I describe this? I would just say, look, hemoglobin has a high affinity for oxygen in high oxygen environments like the lungs. This means it picks up oxygen, or it loads, associates, these are words you can use, associates or loads with oxygen in the lungs. However, in the muscles, remember I said this tells you the environment. You must make the connection. So at low partial pressures, the only place in your body that has a low partial pressure of oxygen is the muscles, because they're using oxygen so quickly. So this is basically the muscles. So we would say, however, the adult hemoglobin has a low affinity for oxygen when it is in the respiring muscle tissue. Okay, that's shown by the low oxygen saturation or low oxygen concentration in areas of low partial pressure, which is the muscle. And then we would say that means it unloads or dissociates with oxygen easily. So you must get the language right and the sequencing right. Now let's have a look at this question now that I've waffled. So, looking at this question, use the information, explain how the seal's myoglobin. Okay, so they got myoglobin in their muscles, kind of purpley colour. Shows the seal is adaptive for diving. Check out the what did I say this is? Is this oxygen concentration? Yes, but when you want to talk about the oxygen concentration, use the word affinity as well. And when you want to talk about kPa, partial pressure, kPa, talk about the environment. So, how would we answer this question? To answer this question, we want to say, right, what can I say about the seal's myoglobin? That's this dashed line. I want to say, ooh, this shows that the seal has a high concentration of oxygen or high oxygen saturation at low partial pressures, right? That's kind of reading the graph. They ain't giving away marks for that. So I need to say this means the seal has a high, its hemoglobin. I'll still make that statement, right? Because they might not be giving away marks for it, but I'm still going to say it just in case. Don't want to punch myself in the face because I missed the obvious, read the graph. But then I want to say this translates into the seal's hemoglobin, myoglobin, sorry, because it's about myoglobin, has a high affinity for oxygen when the partial pressure is low, i.e., far down underwater where there's no oxygen, where it's having to like hold its breath and it doesn't have, it's in a low oxygen environment like deep under the water. So that shows that its hemoglobin is loading with oxygen and not dissociating at low pressures. Something along those lines. Oh, sugar. Touch my mouse is so sensitive. Here we go. Right, I'm going to find the answers. Okay. Yeah. Oh. And you can compare it with, there's the answers. We can compare it. I mean, I didn't really have all of this question down, but we can compare it with, so notice straight away, look. Do they mention oxygen saturation? Do they mention kPa? No. I would still talk about it and add it in, but have a look. They're talking about the environment and look, higher affinity for oxygen. What does that mean? It means it associates more readily or doesn't unload as readily or dissociate, except holds oxygen at lower partial pressures. Okay, they have used lower partial. Allows aerobic respiration when diving at lower, or provides oxygen when hemoglobin unloads, blah, 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 blah. Delays anaerobic respiration, lactate production, except oxygen store, whatever, blah, blah, blah. I don't think I got all of that in there. Yeah, cool. Now, over here, these ones are a little confusing to people with the circulatory system. What we got? Lungs, H. This one, give the letter that represents each of these blood vessels. Aorta. So remember that these are all kind of messed up, but where's the aorta going? It's going to the body. So what do we want? I'm going to say D. We're going to the body. So D. Oh, you can't see D, can you? Because my flipping head's on it. Stupid head. Where are we, T? Okay, so D is the aorta. Why? You're expecting, see where they're trying to F with you here, you're expecting the aorta coming out the top of the heart, that business. But of course that is the atrium. These four squares, that would be the atrium. It must be coming out the left ventricle. It's just they're representing it as a rectangle with four squares. So that is the aorta. Then the renal vein, renal means kidney. So the vein is coming out of the kidney and going back to the vena cava, so is G. And then the vena cava must be F. That was kind of nice, right? DGF. Yeah, H would be, you're right. So H is the pulmonary vein. Very good. Yeah, H would be the pulmonary vein. I don't think they asked about H though, but yeah. Name the blood vessel that carries blood to the heart muscle. Yes, so something, guys, to remember is that here is your heart, and then your aorta is coming out here, then bending down behind the heart and going off, right, to the rest of the body. The highest blood pressure in the whole of the human body is going to be here in the, well, maybe not actually. The glomerulus would be pretty high, right, due to the efferent and afferent arterioles. But the highest blood pressure in like the major blood vessels is going to be the aorta. Every millimeter the blood travels along your aorta, it's losing pressure. So the left ventricle provides the hydrostatic pressure. That blood is going around the body. Remember, the first stop is, oh, I need to feed my own muscles in the heart wall, and we call this the coronary artery. And of course, this is where you get plaques and atheromas, and depending on where it is, decides whether all your heart muscle dies, you just get a mini heart attack or whatever. So that is the coronary artery that feeds the ventricle and atrial walls for the right and the left and spreads out around there, delivering oxygen. Of course, you need to know all your stents and statins crap and all of that stuff as well. But I ain't going through all of that. So, yeah, and the risk factors for that sort of stuff. And anything to do with high blood pressure, like being stressed because you've got exams tomorrow, right, is contributing to you having cardiovascular disease. Because, unfortunately, don't worry, it's a long-term thing. You won't get it for another 25 years. The damage these A-levels will do to you, you won't realise for a long time. But anything that raises your blood pressure and stresses you out roughens the endothelium, roughens the inside of the wall, right? The epithelial layer or whatever. It all gets bumpy, and then it makes more likely for, you know, blood clots to form and all of that, and atheromas to bulge through and all of that, plaque formation, and restrict blood flow to the heart muscle. So anything that stresses you out, high salt in your diet, anything like that, loads of loser LDLs, low density lipoproteins. What do LDLs do? Low density lipoproteins take fats. I can't remember if you need to know it for AQA, but they take fats from your liver and deposit them in your blood, whereas happy DLs, high density lipoproteins, take fats from your blood and they put them in the liver. So they're not in your blood and they're not a risk for CVD. What do we need to do? Yeah, we did mass transport in that. Immunity can come up. Yeah. 2018, Fatima. What time is it? Because I am free tomorrow to do one before the exam, but I don't know. I've got a lesson tonight at 9 o'clock. Someone's having a late night revision, so I'm chipping out at like 10 to 9. Oh, you say lipoprotein stuff, was that old spec? Oh, right, you don't need that then. I don't know. What time's the exam? Is it 1 o'clock? Is it afternoon? Oh, there you go. That means I can do a couple of hours in the morning. A bit of extra revision if it's afternoon. Oh, we've already done all that mass flow business. Oh yeah, that's another experiment. I can't speak. Experiment. What did I use? Methylene blue. I can't remember what they use. Ink. We used ink when we were in school, but they might use like methylene blue or something. Color water up the salts. That's just asking you cohesion tension. So that is just, yeah, you know that business. Cohesion, dipole water molecules, hydrogen bond to each other in a continuous water column. Evaporation of water through the stomata creates tension. Pulling creates a pull or a tension which pulls the water up, and then you just explain the cohesion, and maybe on addition you talk about adhesion to the lignin. Mass flow, understanding of the mass flow, explain how pressure is generated. Oh, this is how pressure is generated in the phloem tube. So that is just saying active loading of sugar in the top of the phloem, water moving by osmosis from the xylem to the phloem because the active loading of sugars lowered the water potential, causing water to move in from the xylem by osmosis. If you want to get fancy, through bordered pits and across the cambium, but they probably won't give you marks for that. And then that creates a high water pressure and water moves down a pressure gradient. What do we want to do? We want to talk about, what have we missed out? Oh, I haven't talked about insects. So, I have got PowerPoints on all of that, but I haven't got them up at the moment. I ain't going to have that kind of time. But guys, all you have to know about insects, I'm going to give you the hard, quick facts about insects, okay? So, yes, there's a dissection we can do for insects. Yes, you'll get marks for saying pin the insect to a dissecting board. Yes, you'll get marks for saying cut away from your body. You're going to remove the outer shell of the insect, which is called the exoskeleton. You're going to expose the abdomen, and you're going to inject into the abdomen some saline solution. Ooh, what's saline solution? It's the same stuff you put in your contact lenses. It's basically a mixture of water with salts in it, salt water. And what that's going to do, that is going to make the tracheal system, which is the respiratory system of the insect, show up as silver lines. And that's all we got to know for the insects. And they might ask you a 1 marker on humane, like get the insect and smack it really hard. No, suffocate it. No, put it in the freezer. I don't know, but there'll be an ethical thing about killing the insects, potentially 1 mark or potentially not. So the tracheal system of insects, what do we need to know? The holes along the side of the insect are known as spiracles. Let me quickly just do this. Will take, let me just spend 60 seconds if he can get a ting out. Let's do 60 seconds on insects, just so you know, in case you get an insected question. Oh my god, come on Tom, fill the thing. Right, let's give you a quick insect down low. Oh my gosh, come on pen, don't fail me now. Right, so OMG, look at that drawing. How many body parts do insects have? I think they have 3 parts. They have a head, thorax, and abdomen. Yeah, they got whatever. Anyway, we got spiracles. This is known as spiracle. They can open and close the spiracle, like if they fall in water and you've ever tried to drown an insect in your sink. If you only wait 5 minutes, it can survive. It can close its spiracles. Now, you do need to know that unlike us who can do this, we can do all the intercostal muscle, ventilate our lungs. How do they ventilate their respiratory system? There are 2 methods. So they ventilate their tracheal system. It's called the tracheal system. And they ventilate it by beating wings and by doing like abdominal movements, rhythmic abdominal movements. Why rhythmic? I don't know. Rhythmic abdominal movements. They basically start doing sit-ups and stuff, or they sit down on a branch and beat their wings. What is that doing? That's creating a pressure gradient. Mass flow is creating pressure. It's increasing the pressure outside the spiracle so that the air goes in down a pressure gradient. Now, let's look at the spiracle system. The spiracle system is made of tubes with rings of cartilage. What do we call these? These have got the most effed up spelling in the world. Tracheae. Trachea. Right? What a messed up spelling. There's an extra E on the end. These are called trachea, and then these break up into tracheoles, right? I hope they're tracheos and not tracheoles, or that's just going to get insane. Right, and then you can see large surface area. So what we're going to say, we're going to say large surface area to volume ratio kind of crap. And these, unlike us, their tubes do not transport oxygen in blood. These do not have blood in them. They have tracheal fluid. There is a tiny bit of tracheal fluid, especially at the ends here where we meet the muscle. They actually go direct into muscle tissue and deliver the oxygen direct into cells and tissues. So they go bang, straight and attach to cells and tissues and deliver the oxygen straight. The oxygen moves, diffuses along the trachea and the tracheoles into the muscles. So you've got spiracle, trachea, tracheoles. Oxygen diffuses down them, but they have to ventilate fresh air in to maintain the concentration gradient. They ventilate it with rhythmic abdominal movements and beating of the wings. That maintains the concentration gradient for rapid diffusion of oxygen along the trachea and tracheoles. I believe there's some sort of fluid there which increases the surface area of contact and crap like that. Ooh. Yeah, apparently there is also a website called Exam Pack, but you have to pay for it. Exam Pack something or other. One of my students had all the 2025 papers. I was like, yo, I'm not allowed to touch them because they're locked, so I can't be dealing with any of that stuff. That's all lockaged. Whoa, sir, what you drawing? You know that drawing was heavy. Look at that insect. It's a grade. That's an A star. That's an A star insect. I think the gases can diffuse through the tracheal fluid. Not that I think you have to say the tracheal. Oh my God, what happened to my beautiful picture? Yeah. There is blood in an insect, guys, just not in the tubes. So do you remember the kind of circulatory system an insect has? It's open, and yeah, it has an open. So the blood is just moving from its heart. It has a small pump like heart that pumps blood through its system, but that blood is for delivering glucose and stuff and other things, nutrients. It's not for carrying oxygen. So unlike us, where the blood carries our oxygen, the insects doesn't. Its oxygen moves through this tracheal system directly into cells, so it doesn't go into the blood first. Ringing. Yeah, I keep forgetting with ringing. Ringing is where they remove the bark at a certain point. I did a live on this recently, and I can't even remember. It was about 4 weeks ago. At my age, my memory is just crap. So what do they remove? They remove the phloem, and therefore, because the phloem has been removed, so they remove the phloem is outside of the xylem, so they remove an outer layer of the bark with the phloem in, and because of that, mass transport can't happen, and the sugars all build up at the top where you remove your ring, so you've broken the phloem, and therefore it bulges, and that just says it bulges at the top, not at the bottom, and that just says that the sugars generally travel from the leaves down to the roots. It's just kind of that ringing. And then the radioactive isotope is slightly different. The radioactive isotope is carbon-14. I have talked about it. And you give the plant a source of carbon-14. Radiation will decolorize like X-ray film. It will make it go white or black. It will change the color, basically. It will on photographic film, like X-rays, it will disturb photographic film. So you can therefore trace the movement of it, and when we trace the movement of it, it goes down. You can say about how you kill the plant, you like the leaves and the stem in different chunks of it in boiling water and whatnot. Yeah, you need a load of different plants, but then you can piece it all together and show its movements. But that's all a bit long. What else? Because it's getting late. It's 8:30. Has anyone got any general questions? I haven't talked about immunity. I might do a bit of immunity tomorrow. Did I talk about cell membrane? I think I talked about cell membrane. You know, you've got glycolipids. There's also glycoproteins. Glycolipids, glycoproteins. Do remember that glycoproteins can be antigens. So proteins on the cell surface membrane can be antigens, they can be recognition sites, they can be for communication, they can cause adhesion between cells because cells like to group together, so they like to stick together and not go floating off away from each other, so they can help cells bind together or adhere to each other. They can also be recognition sites, they can be target sites for hormones, receptor sites for hormones I should say, and all of those kind of things. And they can be antigens, like marking self, whether you're self or you're foreign, like kind of thing. The ELISA test, yeah, I got an explanation on my channel. Hold on, let me just put up a little link in the old Chattacus Maximus. How do I do that? Hold on. I've got an ELISA test. It's long to explain the old ELISA. Now, I have a little video. I've got a series of videos on the ELISA test. Oh my days, what are these ones? I don't know which one is a good one. I've done a question packs, 37 minutes, probably a bit long to be watching, but let me just copy the link. Save it. Oh, no, I don't want to save the link, dude. I want to copy the link, please, Bob. Thank you. Copy link address, bish bash bosh, and then what the fudge am I swear, man, doing these lives I go mental, too much coffee. Right, let's go back in here, back to the old Livicus Maximus Iridius, whatever that means, and then there we go. That's a link to some questions. Hold on, does that work, that link? Oh yeah, it does. Yeah, that's a link to a whole pack of questions on the ELISA test. Somewhere in there, I think if you forward through or watch it on a high speed, it's 37 minutes. You don't have to be watching 37 minutes. But somewhere in there, I do a bit of a high speed thing. No, somewhere in there, I do an explanation. I've got another video on it. I can't remember which one. I've got 3 little videos on ELISA test. One of them is an explanation with my crusty drawings and my incredible art skills. Oh my gosh. Yeah, so I've got lives on all of the different stuff as well. So I've got lives which are much more detailed than the live before the exam where I'm trying to rush through everything. I do have lives that are much more detailed. And as you guys said, I might jump on for an hour tomorrow, so it all depends. What am I looking at? Mass flow? No. No, we don't want to do that. What other things? Yeah, there's the ELISA test. Okay, we've got a virus here. Can you name structures A and B? Yeah, you can use sodium transport. Now, if what you're talking about is how do we absorb glucose, I'll go over that tomorrow because I haven't done that. Digestion and absorption. Because you need to know about the active transport of sodium ions out of the epithelial cells, and then there's sodium ions move back. So that is like very similar to active loading in the phloem, but this time instead of hydrogen ions, it's sodium ions. So very, very similar mechanism, but instead of H plus ions in the companion cell in the phloem, they load, which you don't need to know for AQA, but you do for OCR, they load sucrose that way into the companion cell. But it's a very similar mechanism. You pump out the sodium ions, and then the sodium ions move back in through facilitated. So first of all, active transport using a pump, we pump the sodium ions out, and then they move back in through facilitated diffusion through a protein channel. So protein carrier to pump them out, protein channel for them to come back in, and with that protein channel they drag the glucose with them, and the glucose is going against its concentration gradient, but the sodium ions are doing facilitated diffusion. They're going with their concentration gradient that you created by actively transporting them out. So you created the sodium concentration gradient, then the sodium ion go, oh thanks, we'll go in by facilitated diffusion, and on the way they drag the glucose with them, which is going against the glucose's concentration gradient, and this ensures simple diffusion alone wouldn't be able to absorb a lot of glucose in the 20 minutes or however long it takes for the food to move through the small intestine. So we have to use a mechanism of like indirect active transport, or it's known as co-transport through a co-transport protein. Yes, but I might go over that with a little drawing, because I've got a drawing somewhere of it where I can just add and label it, and it's just hard if I'm doing it without the visuals. So yeah, structures A and B. So B is the capsid. What is A? What is A, guys? Attachment protein. Awesome. Yeah, describe how HIV is replicated. This is quite common in AQA. Describe how HIV is replicated. So they want you to explain, okay, look, if you get ignored, does it matter? So you could say the HIV binds to whatever CD, whatever receptor it is. You're not going to get marks for that via the attachment protein. You're not going to get marks for that. So I don't know, injects the capsid into, or injects its genetic material into the cell. Maybe that's the first mark. I've got the mark scheme, so hopefully. So injects the genetic material in. Then what happens? Once the genetic material is in, what happens? CD4, yeah, you can do. No, not in the absorption of glucose, it's sodium ions. There's no, I think it's pointing to the attachment protein. It'd be great if I had the answers. Do I have the answers? No, I don't have the answers because I'm a twat. What is that? Hold on, is that the answers? Maybe I have got the answers. I'm not really sure. What is that? What if I do that? Is that the answers? Is that the answers? What the fudge? Oh my god, my answers are upside down. Hey, attachment protein and capsid. No, attachment protein and capsid. Anyway, let's move that back. How do I move the screen back? How did that go? I've got the answers on the side here. For about 50% I've bothered to get the answers, and sometimes I don't bother to get the answers. I've got the answers here. How do I move that sugar ray back? Does that work? Oh, if I do that, will that correct? Oh my god, my screen. We're getting funky. Oh my god, what have I done? I've proper flopped hard here, right? I don't know what's going on. Why did I fiddle around? Oh, he's fiddling. It's getting late. The other day I was doing... Oh, yeah, anyway. It's all getting a bit Pete Tong. Right, yes. Let's just put the answers up. Enough of my waffle, because I'm getting tired. Too much talking, 3 hours. So attachment protein, capsid. Attachment. Oh, you could get a mark for saying the attachment proteins bind to the receptor on the T helper cell. Remember, HIV invades T helper cells. They don't specifically give you a mark for CD4, but clever you, put in CD4. Nucleic acid, RNA. We have to be specific. We can't say genetic material at A level. Bummers. So the nucleic acid or the RNA enters the cell. That's if it's an RNA virus. Oh yes, HIV is an RNA virus, whereas was COVID RNA? I can't remember. But you do have to know that HIV is an RNA virus. I guess they kind of show that with the two strands not being bound together in DNA formation. So reverse transcriptase then converts the RNA to DNA, and then the DNA is incorporated in the host cell's DNA, and then we need to say the viral proteins are produced and assembled and then released from the cell bursts, or yeah. Yeah, so attachment proteins bind to receptors on the cell, capsid fuse, insert the RNA. Nice one, you said RNA, Emily. Reverse transcriptase converts the viral DNA, viral proteins form, and leaves the phospholipid membrane, which forms envelope. Okay, yeah. Remember that I'll do some application questions tomorrow. I've got to go and dig up some application questions because I haven't got any application questions as far as I know offhand. ADC are molecules made of a monoclonal antibody linked to a cancer drug. What is this? Use your knowledge of phagocytosis. Describe how ADC enters and kills the tumor. Ew. Oh, that's nice and easy. Use your knowledge of phagocytosis to describe how an ADC cell enters and kills the tumor. So look, just follow it around what is going on. ADC binds to an antigen on the surface of the tumor cell. What is this that's forming? What is forming? What is going to it? What's it releasing? And then what happens there? Nice. Phagosome, you can call it phagocytic vesicle. I guess I'm going to be going about like 9. No, I've got to drop my kids off at school and nursery, so probably like 9:30, 10, and then I'll do like an hour or two. Yeah, so what do we call it? What do we call that? Yeah, so you're absolutely right. So forms of phagosome, whether we're going to say like, yeah, it binds, so ADC binds the antigen on the surface of the tumor cell. I don't know whether you get endocytosis, maybe. And then it's engulfed. It looks like it is engulfed by the cell and then forms a phagosome or phagocytic vesicle. Some people call it phagocytic vacuole or phagosome. All of those are correct biological terminology. And then we've got the lysosome, which fuses with the phagosome and releases its lytic enzymes or lysozymes or hydrolytic enzymes. So lytic enzymes, hydrolytic enzymes, or lysozymes. Zymes, Z-Y-M-E-S, is the enzymes that will digest and destroy the ADC, and that releases the drug, which can then go on and do apoptosis. No? Okay, cool. Might be 1 PA. No, I just went No farting. Sometimes I go. Someone the other day I went. Someone thought I'd farted, but no, it was me going like, oh, look at this graph or whatever. I did it in one of my videos. Someone commented and said, oh, sir, did you fart? I was like, no. I'm going to finish in 5 minutes, but I will bust out some application. I'll do some application questions tomorrow. I haven't done maths. I haven't done loads of things, but you know, what are you going to do? You can't revise the whole year in. I haven't done any biodiversity. Give 2 types of cells other than pathogens that can stimulate an immune response. So other than pathogens, what cells can stimulate an immune response? Log graphs, yes. I've got log graphs. I've got a little video where I go over a log graph. I've got a little video with a log graph. I've got a whole pack of I've got a whole video where I do like maths questions and log graphs and that. Let me find that and lick that in the chat in case you want to have a little watch of that. So while you have a go at these people, I'm just going to find me old maths. Let me find me maths. More maths calculations. I need to blank that. Is this one got the log graph on it? No, that's just loads of maths calculations. Okay, that's maths, zmaths. That's no good to me. Oh, I will get you a log one. All right. I'm coming back. I'm coming back. Right, that is this is a maths video for those of you that want a quick maths fix. There's a few maths questions there on that video, and then let me just hit you with a log. I have got a log. I did a log. No, that's Charning Gold pre-log. Oh yeah, here we go. Log numbers, biology A level. Boom. Yeah, it's better than me teaching it now, like really craply to you. So there we go. This is the log. This last link log video. So there's a log video where I go through a log question. Badly probably, but you know, you can always give it a go. Now, practicals can definitely come up, Tiffany, in paper 1. Practicals are definitely able to come up. Things like colorimetry. Things like your beetroot practical with your cell membrane and then having to do the colorimetry. Yeah, there's practicals. Mitosis and meiosis, you know, so it's mitosis, yeah. And meiosis as well. I haven't gone over mitosis or meiosis, boy. Listen, I'm going to give you the rundown for, let me give you the rundown for meiosis quickly, right? For meiosis, all you have to know, well, the main thing you've got to know, if you have to remember 4 things about meiosis, just remember that meiosis creates variation. I know there's 2 cell divisions and you get 4 haploid daughter cells. That's your GCSE content. But your A-level content for meiosis is during meiosis 1, which includes prophase, metaphase, anaphase and telophase. So meiosis 1, in the prophase of meiosis 1 we have crossing over. If you have to specifically know, it's the prophase. That's where bivalents form and alleles are swapped between homologous pairs, not between sister chromatids. Sister chromatids are identical. Swapping alleles would make no difference. So it's between homologous pairs. They form a bivalent, a chiasmata, where the different chromatids link and they swap their alleles. That introduces variation in meiosis. There's a second way that meiosis creates variation, and that is by independent assortment, random segregation or independent segregation. It doesn't matter the order of those 2 words. As long as you're saying independent assortment or random segregation, it doesn't really matter. That happens during the metaphase and the anaphase in meiosis 1. It actually also happens in meiosis 2 as well. Crossing over only happens in meiosis 1 because there are no homologous pairs in meiosis 2. Remember, the homologous pairs separate in meiosis 1. And then in meiosis 2, the centromeres divide and the sister chromatids separate. But you get variation from crossing over and from independent assortment. Is meiosis in paper 1? I think it is. Anywho, that's the way you get your 2 lots of variation by meiosis, like that. We don't know how they're going to line up in the metaphase and which pole they're going to go to when they split in early anaphase. So between the metaphase and early anaphase, so between metaphase and in the anaphase, you get independent assortment random segregation, because we don't know which pole each chromosome is going to go to. So that causes variation. 2 ways of variation added in meiosis. And then you've got, of course, fertilization, random fusion of gametes, the sperm and the egg. And then you've got random mutations and things as well. Okay. You guys can do an all-nighter, but Mia's not doing an all-nighter. I've got to go to work in. I'm working in 10 minutes. I'm working in 10. So, yeah. 2 types of cells other than pathogens. So here you wanted to say an APC, antigen presenting cell, i.e. a white blood cell, a phagocyte. That's not a pathogen, but it will present antigens. And then number 2 would be a cancer cell or a tumor cell, can stimulate an immune response. Trying to think what else. A host cell to a virus can become an APC, so stimulating it. So like phagocyte and a host cell to a virus. Label the diagrams. X to show where the antigen antibody complex forms. So that, folks, hopefully you know here, this is the antigen antibody binding site. You know that that occurs there. And this is part of agglutination because it can bind to 2 pathogens, clumping them together, which enables phagocytosis to happen more easily. You have a hinge region, you have a disulfide bridge, which holds the quaternary structure together, gives it its 3D shape. That's the disulfide bridge. And then you have a little bit of flexibility here. You've got the light chains and the heavy chains. So there's 4 chains in an antigen. So it's got a quaternary structure, and that will give it its final 3D shape, not the tertiary structure in this case. So you might get some sort of application question about that. Yeah, what is the role of the disulfide bridge in forming the quaternary structure? I didn't even read the bloody question. Yeah, so it holds the chains together, the 2 heavy chains together. And it gives the 3D shape. Describe the role of antibodies in producing a positive result in an ELISA test. Yeah, like I said, I've got a video on that because I am going to shoot now. I might come on for an hour tomorrow and do some application questions. I didn't really go through any of this business either. I didn't go through any of this stuff. So we've got some meiosis going on there. I didn't go through it. Just remember, with your classification, you have a hierarchy of non-overlapping groups. This shows a hierarchy, this diagram. Why? Because in classification, none of the groups overlap. Look, Erecta and Willy, they could have thought of better words than that, right? Erecta and Willy Stoney do not overlap, right? And Drosophila do not overlap. As you go down, the groups get smaller but more closely related. That is why it's a hierarchy. So as we go down the classification system, for instance, we start at eukaryotes. That's fungi, animals, plants, protists. Then we're like, so the first group is really broad, but then you come down into plants and animals, which is the kingdom after the domains. But plants and animals don't overlap. There's no overlap. You're either a plant or an animal. But as we go down, we get more closely related. And then you go into animals and you get vertebrates and invertebrates. There's no overlap between invertebrates and vertebrates, but they're more closely related than plants and animals. So as you go down the hierarchy, you become more closely related. The groups get smaller, but there is no overlap between the groups. Hi. And then you've got to do the binomial naming system. Look, binomial, capital letter, that is genus. No. Oh, no, it's not genus. Excusez-moi. Drosophilidae, and then we have genus, and then we have species, which is all in lowercase, Erecta and Willistoni, or Sex Vitus, these guys are bad, and Neglectus. Yeah, I've had a lot of Neglectus Sex Vitus in my life, but that's another story, right? Species, and that is genus. Notice the genus always has capital. What's the one above that? Then you've got to go king, prawn, curry, or fat, greasy sausages. Fat, greasy, family. So that is family. How do we know that? Because it's the third group from the end. So you've got to know your order. And then bish, bash, bosh. Right, I'm out of here, peeps. I will see you lot later. Yeah. You don't know if they're my kids or not. No, they are my kids. They are my kids. I wouldn't be dropping them off at nursery and taking them to school otherwise, unless I was incredibly generous. All right, guys, don't get too stressed. I will be on for an hour tomorrow.
I can't handle the stress no more, dog. These damn A levels.
Yeah. All right, guys. I do have some predictions for the exams, so don't get yourself worried.
What's your strategy?
Don't need any.
What's your prediction?
Prediction?
Yes, prediction.
Pain.
All right, nice one, guys. I will be on tomorrow, probably about 9:30. I'll be on tomorrow, and I'm only coming on for an hour. Any last-minute requests? I probably won't go through anything I've covered today, right? So I will go through anything that I didn't cover today. I'll do some because people were screaming application questions and graphs, so I'll do that. Check out my video on logs. It's in the chat somewhere, and there's a video on a little video with like 4 maths questions as well. I've got some other videos on my site or whatever. And shout to all the gangsters in the chat. So yeah, I don't have any predictions. Shout to all the gangster bad man in the chat. You know, there's always some people getting too excited in the chat. All right, guys, see you later. Take care. Oh, yeah, don't forget, good luck, yeah?
You're still here?